Gupta for the welcome address.
Hello, good morning. Can you hear me?
Yeah, we can hear you, sir. We can.
Okay. Um, Good morning to all friends, colleagues, delegates, faculty members who have joined here for the fourth ASPIRE workshop. First of all, let me begin by congratulating the organizers of this workshop. And I believe ASPIRE is happening for the fourth time consecutively. It started during the time of COVID, but it has carried on beyond that time point. And I'm given to understand that this time there are 1,400 registrations for the morning lectures, and there were 96 applications for the protocol development, of which I think either 42 or 43 have been shortlisted. First of all, two things. One, that Aspire is fulfilling a felt need among biomedical, medical, and related individuals within this country. 1,400 registrations is a huge number by any standards. So the second thing, that the Aspire workshop has come along some way since the time that it started, and I was more intimately involved with its planning in the first 1 or 2 years since then, and has evolved. A large part of the credit needs to be given to the founding organizers, the three of them, Vikram Gota, Sadhna Kannan, and Jayant Goda Sastri, along with colleagues from other institutions who have brought it to the level that it is now such a sought after conference and workshop. Santam Chakrabarti from TMC Kolkata has also co-organized this year's workshop, and I would like, on behalf of Tata Memorial Center, I would like to thank him for his contributions. I would also like to say that although the scientific content of the conference and the workshop is the real heart of any conference, it cannot happen without the generous support of the sponsor. So it is also very creditable that AstraZeneca India have consistently supported this workshop from its inception 4 years ago to this edition of the workshop. And I would like to thank them for the support. This is well and truly something that will go a long way in building human resource capacity in terms of being able to conduct high quality research within India. Just deviating a little bit from Aspire, some of you will know, but maybe some of you will not, that India has set itself a very ambitious target of becoming a developed country by the year 2047. Becoming a developed country has many different connotations, including, of course, social and economic development, human development indices, measures such as childhood infant mortality, maternal mortality, life expectancy at birth, etc. But one of the attributes of a developed country is the fact that it has planning of this event. And I would also like to thank all the faculty members, and I see there are very distinguished faculty members on the brochure. So my thanks to them for sparing their time, and also to the delegates for their enthusiastic participation. So thank you very much, and I wish you all the
Thank you so much, sir. Now, please join me in welcoming Dr. Kumar Prabhash, Professor, Medical Oncology, Tata Memorial Hospital. Dr. Kumar Prabhash has been a part of ASPIRE since the beginning. We cannot ask for a better start than to hear from him his experiences of ASPIRE over the last 3 years and how does he see the scaling up. Welcome you, sir.
Good morning, everyone. And at the outset, I'd like to thank Vikram for really conceptualizing it. This was really an unmet need where a whole lot of people wanted to have to come together, learn from each other, share things, go back and execute. Yes, my association has been from the beginning because of the kindness from Vikram, and I have thoroughly enjoyed it. And that's primarily because the kind of participants we get, and the way they are so enthusiastic. We think that research happens pretty less in this country. This is the fact of life. There is a very few centers where it really happens. So we get a feeling not necessarily how much people are interested, but when you come back and be in these kind of meeting and the kind of people comes here, then you realize that there is a whole lot of restlessness among younger people. And in our country, we are as heterogeneous as we look, as heterogeneous as we behave when we are in the institution in the chair, not necessarily behaving as a human being, that we are definitely heterogeneous. You will see all kinds of rules floated when you talk to your people in a different institution, and suddenly you realize this is the same country, this is the same city, and this is the same group of people we meet, but there suddenly you realize there is some striking rule is there, and people are not able to do the part they can do as a research there. Then say, hey, this rule, you are allowed to do it, and some other center in the same country, same city, same state is not allowed to do it. So hand-holding beyond that, now I know that will require another mechanism to develop, but that will be very useful, that giving a feedback to all those chairs and authorities that, hey, these rules interpretation may not be the best what they are doing, and that could be hampering the research in that part of that place wherever people go back and they want to implement research. So that will be very useful. That can be part of the future. But again, I need to congratulate and thank both to Vikram and his team for this wonderful meeting. And also what Dr. Sudeep sir said was right. AstraZeneca, they have been part of it. I'm aware of that from the beginning, and wholeheartedly they have supported, especially the part of fund they have kept for the research. I absolutely admire. Usually people forget that part is so important. That makes people attracted beyond only learning. And thank you so much. Thank you so much.
Thank you so much, sir. Next, it's a honor to have with us today Dr. Anil Kukreja, Vice President, Medical and Regulatory Affairs, AstraZeneca India. Ladies and gentlemen, please join me in welcoming Dr. Anil Kukreja for sharing his insights and introductory remarks. Welcome you, sir.
And we truly believe that these life-changing innovations elevate the patient outcomes, enhance the patient outcomes, and improve the better quality of life. As we are a science-focused, science-led company, we thought it is important that beyond our medicines, beyond our products, what else we can do to shape the whole landscape. So one of the identified need in discussion with Dr. Kumar Prabhash and in discussion with Dr. Sudeep Gupta, we identified is to build the research capabilities in the country. And that was the reason why this contribution came wholeheartedly from our side. When resources are kind of required to be spent, we still felt that it is important, and this is an unmet need where we should continue to contribute. So with that few words, I really want to thank. It's a humbling opportunity for us as an organization. Personally, I am very much committed to advancing this research. I strongly feel that this forum, we have scaled up to this level, but there is a huge opportunity to scale it up further. And we are absolutely open for all other, and we were just discussing with Dr. Vikram prior to this over a cup of coffee. How can we make this as a workshop which results into really the scientific protocols coming in different areas of unmet needs? And can we really look at the global evidence gaps where India contributes for the research for the first time and address the global gap sitting out of India? And today, India has that potential, has those capabilities. For that, we need to inspire the rest of the stakeholders to join this movement and be part of this movement to take it ahead. And I'm sure with Dr. Sudeep's guidance and mentorship and colleagues like Dr. Kumar Prabhash taking it forward, I'm sure this will become a movement. We just have to probably put across our brain together. And I was just discussing with Dr. Vikram also prior to this is, can we look at kind of a white paper or publication really evaluating what is the impact of such kind of programs, so that it becomes kind of a guiding principles for other pharma companies and other stakeholders to join this movement as well. So with that, I'll just thank you. I really thank AAZPIRE team and Dr. Vikram, particularly to you and the whole team, all the faculty members. You are truly contributing for the cause. We are just, it's just a token of kind of a sponsorship from is gone from your side, from all the faculties. And I really want to thank the participants for their real enthusiasm, because you are the future which will conduct the research and bring really the evidence which addresses the unmet needs of patients in India, and also to address the unmet needs particularly for patients across the globe. And that's what Dr. Sudeep also mentioned. If India has to be a developed country, we really have to produce the evidence which is at a global level. So with those few words, thank you so much for providing this opportunity. Thanks a lot.
Thank you so much, sir. Next, we have Dr. Mangesh Kamble, Associate Medical Director for Asia, AstraZeneca, India. Dr. Mangesh has been available to us throughout the process, despite being very busy. He has been there for all the meetings. Thank you so much, sir. Please join us in giving a broader scientific perspective. Thank you.
Good morning, everyone. And first of all, thanks to the participants who are here, as well as those participants who have joined virtually. I think this is an academic feast for all of us, and we should actually relish this feast. So because all this context setting and the scientific perspectives have been already shared, but I'd just like to, and it was then we made a commitment from AstraZeneca that no, we are committed, and we are committed to this capability enhancement perspective and to build in the research capabilities among oncologists or young oncologists in India. And it's a testimony today that we are in the 4th year of ASPIRE, and we are not looking back, we are not trying to curb things, rather we are here to explore more and more going ahead. And we want to further scale this up with the interest, with the kind of participation we have seen across India. And of course, India being one of the most populous country now, so we should take this as a blessing in disguise. So more population is more need gaps, right? And how do we address those need gaps? So we should have those ideas coming in. And frankly speaking, the aim and objective of this workshop is the first time research should be seen from India. I work at Asia area level, and I have seen tremendous research coming from South Korea, coming from Taiwan, and these research are going at global level. And somehow, unfortunately, we don't see India on the map, right? And this is one small, collaborative effort we are trying from an academic perspective to ignite this spark amongst these young researchers that no idea should die off, and we should try to nurture those ideas and create those ideation mindsets so that we have some first time research, NCEs, NMEs, phase 1s coming in from India, and India should really shine at a global platform. So that's what I had to share. I wish you all the best, and I would really like to thank Dr. Vikram, of course, Anupama from ACTREC team, Dr. Kalyani and Dr. Priyanka from this AstraZeneca team who were leading ASPIRE initiative. So I think without them, this is impossible. So let's give our full efforts, be very attentive, critical, and participative throughout the 4 days. And I wish you all the best for all the 4 days of the workshop. Thank you so much.
Thank you so much, sir. Next, we have our lamp lighting ceremony. I'd like to invite Dr. Vikram Gota, Dr. Anil Kukreja, Dr. Kumar Prabhash, Dr. Mangesh Kamble, and Dr. Balu Krishna Nair, and Dr. Bangdiwala. Dr. Dinner Savior. Thank you so much, everyone. Next, we have a vote of thanks by Dr. Vikram Gota.
Thank you very much, Anupama. So, when we conducted the workshop for the first time in 2021, there were about 24 participants for the protocol development and about 600 registrations online for the morning sessions. And then the workshop got over, and then there were always self-doubts as to how the delegates might have perceived the delivery of the program, and and what kind of benefits did they perceive and did they actually have. So there was always this self-doubt. And of course, we got a lot of validation in several ways, because a lot of delegates came back and said, it was very good, well organized, and then they benefited, and some of them went on to submit the protocols, and then, went on to conduct the studies and so on. So that was some validation. But in due course, the real validation came in the form of the number of registrations we kept on getting year after year after year. And today we stand at 1,400, as Dr. Gupta rightly pointed out, the workshop. And therefore, I must thank, my first thanks goes to all the participants of the workshop, the morning session, as well as the protocol development team, for keeping the faith in us that, that you get something that, perhaps adds value to your scientific knowledge and acumen. And that belief and faith that you have shown in us is a big thing. And my first thanks goes to all of you, for coming and attending this workshop. And next, of course, I have to thank Dr. Sudip Gupta for being the pillar of strength as far as this workshop is concerned, because right from day one, he felt that, our team of, Dr. Jayant Goda, Sadhana Kannan, me, could actually, deliver the contents, effectively, and hopefully scale it up for the future as well. So that trust was very important back then. It is important now as well. So thank you very much, sir, for keeping that faith in us and continuing to support us. And next, I would like to thank AstraZeneca once again for continuing to support this workshop, and once again, believing in us, because without the funding, nothing would be possible. And the views expressed by Dr. Kukreja and Dr. Mangesh about making India a research hub, that vision is definitely on, and it is very important. And I feel privileged to be a small part of that vision. And so therefore, thanks to AstraZeneca for funding this workshop. And finally, the faculty of the workshop that have kind of been with us, like be it Dr. Balu Nair, Dr. Kumar Prabhash, from the first year onwards, we have had some of the faculty, core faculty, who have been with us, and now the new faculty being added, like Dr. Bangdiwala, Dr. Dennis Xavier, and so on. So they actually make the workshop what it is. And if the students are the protocols in the afternoon. So a big thank you to all the faculty of the workshop. And finally, the foot soldiers of the workshop, and Dr. Mangesh rightly pointed out, without them, so thank you very much to all of you. And with that, I come to the end of my proposing my vote of thanks. And I wish all of you a fantastic workshop, and hope you will take back home something new, at the end of these 4 days. Thank you very much.
Thank you, everyone. I'd like to personally thank Dr. Sudeep Gupta for joining us. We are at the end of the introduction and inauguration. Sorry. So now we'll go ahead with the scientific program. I'd like to welcome Dr. Vikram Gota for the program overview.
We are crunching it to 3 days because we feel that some of the early career oncologists for which this workshop is meant for, they are like either residents working in a hospital, or they've just started their practice, and for them to dedicate one full week for a workshop is not going to happen. So that is the reason why, so this workshop is catered and it is done over a shorter period of time, which is 3 days. And the way the workshop is designed is we have the lecture sessions in the morning. So the lecture sessions are designed in such a way that on day one, we will, like for example, because this is a protocol development workshop, so all our focus is centered around protocol development. So when it comes to developing a protocol, the important elements of a protocol include, getting your background right, stating your aims and objectives very clearly, and then getting the design of the study right, because the design is so critical for any study. And then the methodological aspects, like what should be the inclusion. Therefore, you have talks on how to state your background, how to define your aims and objectives, and so on day one. And then it will segue nicely into the first day of the protocol, where the students will write the background and aims and objectives. The second day, we are going to talk about study designs. So we have phase 2 trial designs, phase 3 trial designs, observational study designs, and so on. And then it will nicely segue into design and methodology for the on day 2. And on day 3, we have elements such as statistics for beginners, then slightly advanced statistics, survival analysis, and so on and so forth. And then a session on how to write the statistical analysis plan. And that will lead to the protocol development, those elements of protocol, which is statistical analysis plan in the afternoon. So this is how the entire workshop is designed. And of course, on the 4th day of the workshop, what we are going to do is we are going to pick 3 top proposals from each of the 3 groups. So of course, I didn't mention that we'll divide the 42 students that we have, candidates that we have, into 3 groups. Just will have a statistician, and maybe a pharmacologist or other research personnel, other people with expertise. So therefore, each of the group assignment for this protocol are also being done based on the expertise available in that group. Like for example, I think I made this remark in the WhatsApp group also, that it would be nice if you have come prepared with some background already. In the sense, if you've done some background, literature search, we have identified gaps, what are the existing deficiencies in standards of care, and how your study is going to kind of fill that gap. If you have written a little bit of that background, it will be really helpful. If not, of course, you make sure that you come up to speed very quickly. And then on day 1, you will present your idea and your background and aims and objectives to the faculty. And the faculty will kind of give their inputs as to whether it is a good idea, or you may want to tweak it a little bit, what kind of tweaks would be necessary, and so on. And you will go back. So each of you, there are about 14 participants, 3 hours of protocol development time, 180 hours. So roughly you'll get about 10 minutes to kind of pitch your idea and get feedback. So each candidate will get 10 minutes to pitch their idea and get feedback. And once the session is over at, let's say, 5 o'clock in the evening, you will have to go back to your rooms. Make sure that you work on the background and aims and objectives as per the directions given by your faculty. And then you have to send it to your faculty, let's say, by 9 o'clock in the evening. Okay? And the Mice Ideas team will create some Google Drive or whatever, where you can kind of submit your protocols. The faculty will actually work, go through your submissions overnight, okay, and give the remarks before the next day, so that when you come for the next day to write the design and methodology part, your background and aims and objectives are very clear. And on the second day, you make a very quick recap of your background, aims and objectives, and then say, okay, because this is what it is, this is the design that I am proposing, and these are the methodologies that I am proposing. And then once again, you will get inputs. You will go back, you will refine your protocol, and submit it on the Google Drive by, let's say, 9 o'clock in the evening. So the faculty will get about 3 or 4 hours in the night. And just imagine, so as hard as you are working, the faculty is working harder because you are only working on your protocol. The faculty will have to work on 14 protocols in each group. So then they'll come back and with corrections to the design and the methodology. And the third day, you will do the same thing. You will develop the statistical analysis plan, submit it by 9 o'clock, and the following day, they will come back with the comments. So that at the end of, by Sunday, you should have your complete protocol written. And of course, if you go to other workshops, they may also help you to design CRFs, they may help you to design ICFs and so on, which are extremely important elements of any research dossier. But keeping the time constraints in mind, we have to focus on what is the meat of any research study, which is the protocol. So this is how the entire protocol development session is planned out, and this is how the entire lecture series is planned out, so that one will actually lead to the other. And what I want to emphasize here is your cooperation in kind of completing your protocols writing in time and submitting it back to your faculty, so that you have to be mindful of the fact that they have to correct about 14 protocols. So if you are late, you may or may not get your feedback on your protocol. So therefore, please make sure that you stick to the timelines and try to submit it not later than 9 o'clock or latest by 10 p.m. Each day. So that is the overview of the program. And of course, you might have seen the brochures, and in fact, the program is also put up here on the standee. And you will see that some really exceptional people are helping you, develop your protocols, and you can be rest assured that you're in safe hands. But my only request is please cooperate and make sure that you submit your write-ups well in time. Giving adequate time for the faculty to give their feedback. So that's about the overview. Thank you very much, and we'll start with the actual program now with a talk by Dr. Kumar Prabhash.
सर अभी हो रहा है सर 10 सर इतना टाइम 40
So, good morning, everyone, and once again, thanking Vikram for this opportunity. And my job is to talk about how to have the big idea, and how to have that big idea which can ignite people and you to take it forward. So before that, what I think so Vikram was saying, the key was that if you succeed, this meeting succeeds. So key is that you need to succeed to ensure that complete things that will be very useful. And as early you complete, that then you will have another struggle when you go to your own institution to complete things. Coming back to the ideas, so I'll say don't focus on big part. Big has got limited impact. Do you have a good idea, implementable idea or not? And I'll show you some of the example which I thought I had a big idea, and it was, then I realized later on it was not so big. And also I had some not so big ideas. It was just that in the OPD I had something, hey, this is a problem I am seeing in my OPD. I need to have some solution. And then say, hey, giving some solution, and then you realize, okay, it can be meaningful for people all around. Starting with some of how to not have a big idea. And I won't show many of them. I have got many of them, which I have failed, because this is also a self-promotion activity for me. So I'll be showing only some of them, how you fail in having some big ideas. So we started with, and this was a randomized trial we had put. And long back when I joined, I'm a fairly elderly person, so I joined in 2004. And that time letrozole was absolutely new. Many of you will know who works in oncology, especially med-onc part of it. Otherwise also letrozole is a pretty famous drug. And we saw that in lung cancer, you have ER, PR and AR positivity is there. we say, oh, we have got some really big ideas just that time, and we know that in breast it works very well. We say we put a randomized trial, which we had we didn't know the whole lot of biology part of it, but that expression part we knew it, that data was there. We got IC approval, and funding was there for it. And that time, the company which is to make it, they were keen and they said, hey, we will fund it. We didn't know many of those parts. Good scientific basis, got project IC approved, had funding, never got DCGI approval because this was a totally new drug in new place. And that time DCGI was not what you see today. But for any new indication, you have to go to that place. So I fly twice to the Delhi and try to see that if we can get approval. We never got it. So project never started. So otherwise, scientifically looks great. But where I floundered, I never realized what will happen after that. So I may have the earth-shattering idea, but if I'm not able to implement, then there's a problem. My friend in Australia, he did it. His name is Dr. Nimit, and he had a combination, letrozole plus something. And thankfully, it didn't go through. Sometimes you feel at the hindsight it is good because that was a negative study and there were a lot of side effects. But having said that, the key learning was not that. Don't take this learning that, okay, if someone fails and you say, oh, good, yeah, I didn't take this idea. Learning for me was, don't miss this part of it, that what are the logistics for your questions to be implemented. And that should be in your mind, which you will know talking to people around. Was this only one more I'll show you, not more than this. This 2 failures is enough. So this is again, a good idea. It was the paper was in Science where the animal study was there. That was the basis to take it further. And we thought in human, when we do, easy one because you have to do only in the blood, CD26 expression, and to see that what happens in BMT patient, that is hematopoietic stem cell transplant patient, how is the engraftment. So good idea based on animal studies in Science. It went to Annals. Annals is a fairly good journal, and we were very happy about it when we completed. So here it took off. So the project took off. Not only it took off, we completed with the help of my friend here, Dr. Jyoti Kode and Dr. Naveen Khatri. But after that, we realized, okay, it's a good idea. How does it benefit my patient? So and that doesn't mean, so there are all kinds of researchers who wants to work on Golgi apparatus. Why not? Absolutely, they have a right to do it. But I'm a clinician and a bit of a bent of mind that how it benefits my patient. So here it was, okay, how does it benefit my patient? So because we thought of that we do it and we get some CD26 inhibitors. Fairly young, you can see it was long back. It is, I think so, 2010 it got published. We started before that. We started in 2006 or 7, something like that. Then we realized, hey, it's not easy to get these 2 CD26 inhibitor. You don't get easily. It's not like it's available somewhere and you simply ask people, and or if you ask people, they will simply make it because I am requesting it. It doesn't happen that way. So what happened? Good idea. Completed. Good looks in my CV. I put in my APAR, we call annual appraisal. That looks nice. But then we realized we couldn't take it further. Means that it is never going to reach any patient. And that was a bit disheartening. Okay, ideas can look good, but it may not end up benefiting my patient. So it was bringing you to ground. So I've showed you 2 failures. Now not many, not more. I'll show you some other things which was very simple. And also showing something very simple. So I'll show you what we did. So this is pretty common, and we used to see very commonly in our clinic. Big tumors, can't do much. Your surgeons are not uncertain about it, to do surgery or not, to give chemotherapy or not. And we used to get a whole lot of people for chemotherapy at that time in our clinic. And then we started having a discussion. How does it benefit our patient? You give it. How do we know it benefits? Do we have? There is nothing in any guideline. Anyone is aware? Neoadjuvant chemotherapy in any NCCN guideline? So none. We say, why we keep giving it? So that question kept coming. Because that kept coming, we say, hey, we need to work on it. So what we did, we did, no, okay, this is the pay. Okay, so what happened to these patients? What it means, all those 100 patients, therapy, if we use this approach, you can have half of them going for surgery. We say, can we do something more? And we were looking for it, how to develop the less toxic treatment. And we this got opportunity in a place where you can't give DCF. This is a place, Muzaffarpur, which we have our center. We don't have a medical oncologist there. We say, can we have a drug and a combination, which you can use it there, which is less toxic and maybe with a similar response rate. So we call it 5-drug combination, 3 of them given orally, 2 of them given injectable. We call it paclitaxel, platinum, methotrexate, celecoxib, erlotinib. Don't get in details. In case you are interested, please go through it. And looks like response rate wise, it is good. In a bigger cohort, it looks like it is doing good. Again, in the clinic where we had an unmet need, where patient can't take DCF. So you need to have some safer protocol which you can deliver. And at this moment, we are running a randomized trial comparing DCF versus this combination. And if it found to be non-inferior, again, it provides the answer to a patient in the clinic. If you don't have bed, you can't have a PIC line which needs to be maintained. You are not fit for DCF-based combination chemotherapy. This can be a good answer. Again, a small improvement, useful one. Now, does this kind of work impact? So this question remains. डॉक्टर कुमार ये तो सही है, ये तो आपकी अपनी डफली अपनी राग है जी, आपने कर दिया उससे, लेकिन फर्क क्या पड़ा, आप कर रहे होगे? Do you have that people think it is useful? So sure, what we do is we do talk to our colleagues. What do they think when the clinic? So you have NCG guideline, NACT, borderline, all the 15 odd papers or more you will see, primarily you will see from TMH and our group, primarily with this concept. Now we started seeing some more. So we saw some from Kidwai, some from Kashmir. So it looks like people started picking up, and NCG guideline recommends based on the same evidence that this is the way to do. ICMR guideline again picks up that it is way to do. This is the ICMR. This is another guideline, and slowly we say people do pick up because it is meaningful in the clinic, and people start using it. We did one survey, not by me, by a radiation oncologist in Bangalore, and say 6 or 7 out of 10 oncologists, when they have this situation, they use this approach in our country to manage this oral cavity cancer patients if they get this technically unresectable patient. Highlighting that simple things in your clinic, and if you provide a solution, it can be meaningful for your patient. Can these kind of simple things can get you reference in NCCN? Yes, there are times when you think retrospective studies are useless. Highlighting here, and again, ideas, as I told you, I'm self-promoting myself, so don't get me wrong. That is one of the idea here. And you can see this our paper. This was in what? Sinonasal tumor. Retrospective analysis, and you can go back and check NCCN guideline. You have a reference, and this is the basis, one of the basis for using neoadjuvant chemotherapy in this setting. Again, I keep emphasizing, this is a retrospective analysis based in the clinic. They were patients who was not upfront resectable, and our idea was, if that is the case, use chemotherapy, but use in a structured way, and then have it. What happened to those patients? So we didn't use it just like that. We used it in a structured way, and then also analyze what happens to those patients. So highlighting that so many times, simple ideas in the clinic, if you do it, you can give good solution to your patients and the colleagues. One of the thing is affordability. That is always a problem. And sure, if I say that those immunotherapies and cetuximab, less than 10% of your patient can take it. Am I making a fair comment to oncologists here? Okay, so that is fair comment. So this is always a concern we remain for us, that how do you provide a solution to our a patient where they don't have, and that can be cost effective for them. So this is one, no, we knew that once we had this, I don't have to say it as you suggested, that our data suggested less than 3%. Sure, you have a corporate hospital, it can be far higher, but by and large, this is the number which I suggested. So we were keen in developing how do you develop a treatment which is useful for our patients. So this was a metronomic chemotherapy. We have a place called Derwan, it is a few hundred kilometers from away. And my senior and a very dear mentor, Dr. Banavli, he looks after that. And he's a big fan of these things that it's good you are giving it here in TMH, but what happens 200 kilometers beyond TMH? Are you able to give those treatment? And he'll keep nudging us, that if you're not, then you're not doing a good research. I say, हाँ ये बात तो सही है जी, बिल्कुल अगर majority लोग नहीं ले पा रहे. And he is a big fan of metronomic chemotherapy, and with him we learned, hey, can we explore it? We say, how do we explore it? So first patients, they got this treatment because in that place you can't take any other treatment. You can't take injectable there because that fund is not there. So what we did, methotrexate, celecoxib, which are very cheap. Your oncologist here will tell you it comes in few hundred, and people can take it easily. And we say, looks like there is some activity. So you can see here 20 patients data, 60 patient data, which we initially published, which reassured us that it is meaningful in this situation. And this led to our first phase 2 randomized trial comparing IV chemotherapy versus oral metronomic chemotherapy, and suggesting that can you remove IV part of it with head and neck cancer? It will be easier for them. They have to come to OPD, don't require daycare. One person is anyway hassled about daycare here for waiting list. We say that can be removed, and primarily it's easy for patient. Looked in phase 2, this was a thesis of my first student, and it looks that yes, it controls the disease better, it improves survival. And then we did a phase 3 trial because we wanted to reconfirm it before saying, hey, good in bigger number, see that it is really useful. And this showed that yes, it is non-inferior to IV chemotherapy, maybe a bit superior. Also, quality of life is better, and we use it routinely. I'll come to that, where are the places it is recommended. And then you realize it is not in Lancet, main Lancet, it's Lancet Global Health. One can see the impact factor. That is again to self-boasting. Primarily was, we didn't know that it will go to Lancet Global Health or wherever. It went to ASCO oral presentation also, but that was not the basis for doing this. Basis was that hey, IV chemotherapy is a problem. Can we get something oral? Is there some activity? Can I see those drugs which works? We say, hey, if that is the case, let us explore it and answer that question in a way which is useful for our patient. So what we answered, good, you can take it if you're taking IV chemotherapy. Good, it may be useful for your patient. You have some good responses and quality of life can be good. But as I suggested, I use the word some responses. The responses were around 12% odd, not good for our patients. Say, can we make it better? Sure, world was working on cetuximab, we'll come to immunotherapy also. We didn't have that access. We say, can we do something what is available? So what we did, we added erlotinib to it. You say, celecoxib, methotrexate, and we say, what happens to them? There was some, and based on what? There was a data on neoadjuvant chemotherapy in head and neck cancer for erlotinib, where it was 30% response. Say, hey, erlotinib is easily available. Anyway, patients, when they fail, there's nothing. If you give erlotinib, what happens? And in some of our patients, we saw there is some good responses, and that reassured us there is a merit to it. But when we developed, what we did, we say, hey, develop in a way that you combine 3 drugs, which is less toxic, and there is a scope for adding something else. Now, how do you do it? The way to do is reduce some of the doses. So someone can argue, did you reduce doses for all drugs? So remember here, the methotrexate was 15 milligram per meter square. Here it is 9. So we did a phase 1, phase 2 study, not escalating dose, de-escalating dose. And we say, is the outcome similar? And we found outcome can be similar till 9. Below 9, outcome was inferior. We want read it. It's a very nice design to use in a trial in case you want to use a de-escalating dose in your trials. Someone can argue, okay, can't you do for all the drugs to de-escalate? That would have taken few more years. We say, no, one is enough. we need to move on, beyond that, and that's why we moved on from here. So we completed this trial. Important was 40% patients responded. Anyone who manages head and neck cancer will tell you that head and neck cancer, platinum refractory patients, usually half the patient responding to treatment doesn't happen. The responses were short term. There was a problem. But it suggested to us it is meaningful and a good palliation and also cost effective. Again, the cost in TMH doesn't go beyond few hundreds per year, per month. Question come after that. We have done 2 randomized trial. If you are interested, one of them was in ASCO last year as a part of CRSF initiative, phase 3. We took it up and it is in the process for publication. Another was in ESMO as a poster one was by Rishabh, other one by ESMO was by Dr. Akhil, and suggested that if these 3 drugs we gave, it is better than IV chemotherapy in progression-free survival and overall survival both. I'm not putting it here because there are too many slides. Vikram will get upset ki kitna time lega. So you want, you can look towards it. Now say, iska fayda bhi hai kya? As I suggested, how do people use it? So again, I say, I do talk to my colleagues around, do they use it? How do they feel about it? They do use it. And you can say again, National Cancer Grade Guideline, it is there. Up to date, again, this is the same reference you will get. You can check back the reference that people do pick up if you do a good job, which is meaningful for your patients, and they do come in the guidelines. We had, as I told you, we also learned that you don't have a sustained response. And it was in our mind that we need to do something about it. And immunotherapy was costly. That also we knew. And I showed you only 3% of our patients can take it. And we also were reading about it, immunotherapy, and we realized that can we do something with dosing? So one is that how do you decrease the cost? Either company reduces the cost. Now, company is not going to listen to me. AstraZeneca is not going to listen to me. That was on the lighter note. So they don't, because they have their own reasons for it. But can we do something? And we have a wonderful pharmacologist here. And I have a few wonderful pharmacologists here. And Vikram being a dear friend, we take this, in this context, we take a help a lot. Okay, what do you think? This is dose proper. Then he does some modeling and gives, Vikram does, and it's very nice to see those modeling what he does for, drugs and other things. You see, do you require this dose? And then we realize, hey, there is a various reasons to know that that dose is not necessarily required. So we use 1/10 of the dose. It was very obvious. If you use 1/10 of the dose, if it is positive study, the cost will be 1/10, because that time the cost used to be around 2.5 lakh rupees. So that will be benefit the patient. So we did a randomized trial. Overall survival was better. Cost did go down. It was pretty obvious. It was not we have not done economics analysis here, but it is obvious that the cost went down, got discussed at many places, and NCI covered it, and NCI invited also. Again, I say there's a bit of a self-promotion, so you have to bear with me that, it covered that it is useful, and, a whole lot of people in country, they do use this, combination. We keep having discussion with my colleagues that how do you feel about it? Sure. Some patient, just now update will be putting in public domain now. That's a 3-year survival data. So almost 20% patient, they remain long-term survivor. They used to be around 3 to 4%. So this is a good, it is better, not excellent. Sure, you can say ultimately, Dr. Kumar, 80% to phir bhi nahi bach rahe. That is true. But there is a good improvement from what we had earlier. And most important, now majority of our patient can afford it. Some of the things what we do is providing some solutions in patient receiving chemoradiation head and neck cancer. There was always a question, and again, simpler questions. As you might have observed, we go for some simpler questions. So we say, cisplatinum, we used to struggle, I struggled for almost 12, 13 years. That is weekly as good as 3 weekly. We kept reading, and then thankfully, now there are 3 trials in the world, 2 from India, one from TMH, one from AIMS, and which by and large tells us that there is a good answer to it. So this was the first trial, first one to get published in the world, and it showed that weekly with 30 milligram is not good enough for local control. And again, it gets cited at multiple places. Simple question, but we also have a conclusion, evidence from that 40 milligram is as good as 100. So if you're using 40 milligram is good enough, and that was by Dr. Atul sir from AIMS. Then we say, people who can't take cisplatin, what to do? And you have a cetuximab for it. Again, I say 3% people can take it, but again, emphasizing, also learning also teaches you many things. That for cetuximab, there is no data in cisplatin ineligible patient, and that we learned while reading a bit. I say, hey, docetaxel is there. There's some decent data for it. We say, do a randomized trial, see what happens. Anyone can afford it, docetaxel, 15 milligram per meter square. Cost is again 1,500 to 2,000 rupees for whole treatment for docetaxel. And person can say, oh, chemoradiation ka toxicity ka kya hoga? That is a separate one. You need to add some cost to it. And it showed that yes, survival is different. If you use in cisplatin ineligible patient, docetaxel, simple drug. And also, if you see the NCCN now, this is reference there as a reason for docetaxel as an option in NCCN guideline. Targeted therapy is some local solution. In lung cancer, there is a drug called osimertinib. Very good drug, excellent one. Very easy on patient. Majority of our patient can't afford it. So at the same time, when that drug was there in coming to market, we had this protocol running. Combine geftinib plus chemotherapy. This is an older drug. No one wants to care about it. And we found that then you can have a bit more toxicity with a similar outcome for the patient if they can't take osimertinib. And again, I repeat, 95% of our patients can't take osimertinib. Again, I say, does it has a value? Just showing that again, I say, cell proportion. See it, if it inspires you, sure, do all this kind of work. That yes, you do get cited, up to date, ESMO guideline, and many other places you get cited that this drug is useful. This work is useful. And again, highlighting that out of multiple options available, there is many more beyond that now. But something which is affordable is this one for our patients. So any patient can take it if we can give this treatment to our patients. We do some work in molecular part of it, and because you do work with targeted therapy, and there is also effect that if you do one work, you are able to do many more. And you can see here, because we did molecular work, we did randomized trial, we had the testing which we developed in the lab, and then we were able to do many of those things. What happens to double mutation? What happens to ERBB2, which we had a discovery in our lab? Then what happens to squamous cell carcinoma and EGFR mutation? And this way we were able to do because there was one or two work happening, and that gives flip to many other things for yourself. So from my side, developing a research question is one of the important aspect of research. It is important for a question to add value in some aspect. Remember that value, any little value is important. If it treatment which makes a patient stay in the hospital 1 hour less, it is important. If it requires a pain score going from 8 to 4, it is important. So a small little any value for patient, then any of the research is very valuable. Apart from that, I do have my basic researchers who disagree with me on this, and I totally agree with them. Not necessarily. Many times basic research, when we see, in case there are people with the basic research interest, we are not able to see today. But then he gives me example, many other example that people didn't see the research which is happening today, and 20 years down the line, what is the impact? So if someone has those kind of mindset, sure, that is also a good thing to do. It is important to be careful in scientific aspect of research question development. So scientific, if you want to show a benefit, that part will be important. So whenever you want to do a research work, you say patient benefit, be careful what you are using to show that patient benefit. That becomes important. It is equally important to ensure that research question is developed within the resource available. And I keep saying this Hindi, saying that नॉर्मन तेल होगी ना राधा नाचेगी. तो देखो ये तो हर जगह है. तो you have to ensure that कि within the resources, you need to find out, do we have a question which can be answered, and then it will be fruitfuling. And then you realize sometimes resource start getting stretched. There are people who are willing to come and share resource with you, which you had not thought of it. The low dose immunotherapy project, with all the support from hospital, the funding came from a patient. It was a patient whom I used to treat, and she was very inquisitive lady, and she was getting osimertinib, and then she used to tease me कि डॉक्टर साहब इतने महंगी महंगी दवा लिखते हो, नो, तो आई यूज्ड टू, एंड शी वाज वेरी, तो वन डे ऐसे, देखो मैडम ऐसे बोलते तो रहते हो आप, तो ये सच्ची बात है, मैं लिखता तो हूँ, आप भी ले रहे हो, ये बता दो, कुछ हेल्प ही कर दो, हम तो रिसर्च करने को राजी हैं, वी वांट टू गेट सम कॉस्ट इफेक्टिव ट्रीटमेंट, इसे को बताओ. Then that time just now I had IAC approval. Say कि देख लो. Oh, absolutely, she was damn smart lady. She went through everything, and she did realize, and we got rejected from many places for the funding for that, project. And she was like, "Why someone is not sponsoring you?" So I put her, why don't you sponsor if you think it is so good? I say, I'm a researcher. I know if I'm involved, I have all excellent project. But if you also think, fund it, and she funded almost majority part of the research, highlighting that many times if you keep doing this work, then you do realize that from unexpected quarters you get people who end up giving you resources just on the faith of that I am doing a good job. I don't know why she had to give that resource, except that she thought I am doing a good job. We days, my lab where I work with, and if you see all the females, that's primarily because beti bachao beti padhao, so, and my dear mentors, my teacher, and my head of department here, Dr. Banavli, everyone knows, he's academic director, he was my ex-HOD, and our ex-director TMC, absolutely wonderful people, because when you do research, you require people around who trust you and say, hey, you will get in hot waters many times. You require someone around you who say, hey, no issue, please keep doing. And I had those people who were, and lot many people who worked with me. Thank you so much, and happy to take questions. Time overshoot तो नहीं किया मैंने, थोड़ा कर, sorry. So you want to take questions later? So I'm okay with it.
Good. Yeah, thank you, sir. I know, thank you for lucidly and candidly sharing how questions came to you and how you have answered them. And I know from experience that you've been finding questions that are relevant to India and then answering them. And also beyond that, you've inspired many oncologists in India who have actually pursued the same research that you've been doing, and we are getting new answers from the country. So let me take some questions from the floor, maybe at least 2. And if there are something online also, we'll take. Yeah.
Thank you, sir, for that beautiful presentation. So as you said, sir, you have a team in place. So when you started early research, so that team may not have been there. So probably you are alone in doing things. So do you think that in a center where you don't have a lot of people who are combinedly working for research, you will get results if you just focus on one cancer, or you have to find questions and answers for all sorts of cancers? How is it, I mean, good?
Definitions, certain technical areas where we need to be clear as to what we are writing, because very often it's not just about properly articulating your research, it's also about making it follow a set pattern so that the person who's reading your research knows where to look, knows exactly what you're doing, and feels excited enough about your research idea so that they want to fund it, for example, or they want to participate. You might be sharing your protocol with colleagues in other hospitals, and they should feel that this is meaningful research. Now, I noticed that in the sequence of lectures, this lecture comes before the discussion on background and objectives and hypothesis. I feel maybe from the next time onwards, that lecture should come ahead of this lecture. So that lecture will be done next by Dr. Supriya. So I'm just going to focus on the objectives and the endpoints. Right? So, when we frame a research question, we usually start with a hypothesis. You may be feeling that something will make a difference, or something doesn't make a difference, or some, increasing or adding a new agent or increasing a dose makes a difference, for example. And then you want to, set out to write out the aims and objectives of the research program. Now, the hypothesis and aims will be tackled by Dr. Supriya in the next lecture. So what we're going to do is just go over them very, very briefly, not to overlap too much, and then jump into the objectives and endpoints, which is my directive. Now, what is the research hypothesis? Supriya will, I'm sure, discuss this in greater detail, but it's the expectation or the belief with which you are doing the research. I'll try to keep my slides as non-technical as possible, give you as many examples as possible, just to make sure that you understand what each of these are. So, for example, I'm, just taking the cue from Dr. Kumar Prabhash. He felt that his hypothesis was that metronomic chemotherapy is going to lead to, a good outcome. He felt that metronomic chemotherapy will help patients with advanced head and neck cancer. And that was his hypothesis framed in a non-technical way, right? But in a more technical way, when we state the hypothesis, it is what is that the trial wishes to establish. That treatment A can be safely administered in disease X, or treatment A is active, leads to responses in disease X, or treatment A is superior to treatment B in terms of overall survival, or risk factor B is associated with disease Y. Sometimes we don't do research with interventions. We may be looking at prognostication research, or biomarker C is associated with response to treatment D. Now, these are all situations where you have an idea and you feel that that is true. And what is that? What you're trying to do through the trial or the research protocol is trying to establish whether your hypothesis holds or doesn't. Now, once we outline the research hypothesis, we have to write the aims. And again, Supriya will tell you much more in detail. But what is it that your study aims to do is what you write over here. For example, some examples would be that you want to establish the safety of drug X in disease Y, or establish the efficacy of drug X in disease Y, or you want to decide or check whether the drug X improves the overall survival in comparison to the standard of care. Yeah. Or you want to determine the association of certain risk factors or biomarkers. So that is how you frame the aims of a study. Now, when you've done framing the hypothesis and aims, you now have to come to objectives. And sometimes it is really confusing for researchers to, what is the difference between aims and objectives? Don't they really mean the same thing? But there's a slight technical difference, and this is what I want to highlight to you, is that the objectives are a bit more specific compared to the aims, right? A more specific goal of research. How are you going to achieve the aim? What are the, measurement instruments that you're going to use? What are the endpoints that you will measure? That is where the objectives are far more specific than the aims. And usually they are listed in a certain order. And this is an order of importance that you are trying to look at. So let's get down to a real life, or, well, not exactly real life, an imaginary example, but an example. Okay, so let's imagine a fictitious drug. This is a new immunotherapy agent, and it's called zabardastumab. Okay, now I know that immunotherapy agents evoke mixed responses. Some people consider them to be too expensive for the common individual. Some people feel that no, given in low dose, it may be very useful. But let's for a moment imagine this agent, right? So how are you going to go about investigating this agent in its life cycle of research? Now, brief revision of the clinical trial phases. I'm pretty sure all of you know this already. So there are basically four main phases of clinical trials. In the first phase of phase 1, you assess whether this drug is safe. So you assess the dose limiting toxicities and you determine the maximum tolerated dose, right? Once you've done that, once you've got a dose that is safe to use, you then transfer it to the phase 2 clinical trial, where you're basically looking at activity of the particular drug in a particular disease situation. So you assess the activity in a particular disease, and of course you also, in the process of testing it, confirm the safety of the drug. Or for example, whatever intervention that you're looking at, it could be surgery, it could be radiotherapy, it could be, anything. The third phase is, so then, you have assessed the activity and the results are coming out. The results could come out in two ways. It could be discouraging results. For example, the standard of care leads to, say, a 30% response rate, but your drug, which you thought was going to be very useful, gives a 25% response rate, and you don't believe that, this is worth investigating any further, and you might drop the further research. But on the other hand, you could get a drug which in a phase 2 study has, say, a 40 that yes, now is the time. Now that I've got these results, I'm going to test this new drug versus the standard of care in a randomized controlled trial. This is a phase 3 trial which compares the clinical benefit of a particular agent versus the standard of care. Now, let's say that you again can have two kinds of outcomes. You might find out in the phase 3 trial that your drug is actually not better than the standard of care. But on the other hand, you may be successful and the study might show that there is a benefit based on what you set out or what you designed. And this drug then receives regulatory approval. Okay? Sometimes when we are dealing with surgery or radiotherapy, it becomes widely established. It doesn't need regulatory approval maybe, but it might become established and go into a clinical guideline, for example. But once it is in the process, you then also have to do some further research. And this is pretty standard for new drugs, which is a phase 4 clinical trial. And the phase 4 clinical trial verifies whether the clinical benefit obtained in the phase 3 trial continues to be present in the real life setting. And why is that important? Is because in a clinical trial, you often have some restrictions and ineligibility criteria, right? You don't want to try it in an 85-year-old person. You don't want to try it in somebody who has some cardiac disease, etc., etc. But when this drug is approved, you may want to try it in certain situations in a fit 80-year-old person or somebody who has mild cardiac disease. So you really want to know whether in real life the benefits and the toxicities are both okay, right? So that is a phase 4 clinical trial. Now, of course, why are we discussing the phases of clinical trials? Is because our objectives and these different phases of clinical trials are different. And depending on what kind of research you are going to design in the next few days, your objectives and endpoints will have to match what the phase of clinical trial that you are trying to design. So let's take an example, right? So this is the drug that we have designed in the lab. So Dr. Gota, for example, has designed this drug in his lab, right? So, but now he can't just start using them left, right, and center. So he has to design a phase 1 drug trial. And that usually comes out after animal research is done and the research in animals has been presented and it meets safety criteria, and then he wants to try it in first in human studies, for example, a phase 1 drug trial. Now, what is his hypothesis at that time? His hypothesis in a phase 1 trial would be that the single agent of this drug is safe in recurrent or metastatic non-small cell lung cancer and achieves plasma levels that can be tested in a phase 2 study, right? And his aim might be to determine the safety and tolerated doses of this drug in advanced or metastatic non-small cell lung cancer. So with these hypotheses and aims, what are your objectives? Now, as I mentioned, aims and objectives sound similar, but objectives are far more specific. So what are you going to write in the objective section? You're going to write is that our objectives are to determine for this drug the maximum tolerated dose, the dose limiting toxicities, the adverse events based on a specific instrument. Here it's the CTCAE version 5, for example. And of course, looking at we want to determine the pharmacokinetic and pharmacodynamic properties of this drug, right? So these are in order of importance, what you are going to do, right? Those are your objectives for this study. Now, let's say you found the maximum tolerated dose and the dose limiting toxicities, etc., and you think that your drug is good to go to the next phase. So in the next phase, the phase 2 drug trial, your hypothesis and aims change. You want to now know whether zabardastumab is active in recurrent or metastatic non-small cell lung cancer. And your aim is to determine the response and also to verify the adverse effects of this drug in this setting. Now, what then will be your objectives? Okay, so you've seen the aims, what will be your objectives? Your objective will be to determine the objective response rate using a particular criteria. Yeah, your second objective would be to determine the adverse events using a particular criteria. The third objective would be to look at the progression-free survival, although that's not your main goal, right? This is a phase 2 drug trial. Here you're not trying to look at long-term outcomes. You're trying to look at much more immediate or medium-term outcomes. You're going to look at the progression-free survival and the health-related quality of life, for example. You want to check and see what's happening, right? Now, all of these objectives may not be present in this. You could just restrict them to the first 2 or first 3 of them. But these are just examples for you to consider. Now, let's say that you have determined the objective response rate and you find that this is encouraging and you want to go to a phase 3 drug trial, right? Now, this is a phase 3 study, and now the hypothesis and aims have changed some more. So now the hypothesis is that this drug improves treatment outcomes compared to cisplatin and pemetrexed in metastatic non-small cell lung cancer as first-line therapy, or to determine, and the aim will be to determine whether this drug improves treatment outcomes in first-line therapy compared to the standard of care through a double-blind randomized trial. So what will be your objectives here? So your objective will be to compare zabardastumab with cispem in terms of the overall survival, progression-free survival, adverse events, health-related quality of life, and association of, say, PD-L1 levels with PFS. Now, your objective is to compare new agent versus standard of care with certain specific parameters. That's your objective, right? So now that you've defined your objectives, you now have to be even more specific regarding what exactly are you going to measure. And that is where we come to a very, very important topic as to what do you choose as your endpoints in a clinical trial that you're designing. Now, what is an endpoint? An endpoint is something that is associated with your objectives. What are you trying to measure for a particular objective? If it's survival, your endpoint may be overall survival. Okay, if it's a response rate, then what exactly is your endpoint? And the endpoints have to be carefully selected, because what you're basically trying to do is find out what is meaningful either to the patient or in earlier stages of clinical trials to scientific research to decide whether it's going to be worth testing it on the basis of whether it's meaningful to patients or not. And your endpoints have to be, parameters which can be measured unambiguously. There should be no confusion. It should not be, somebody's impression. It has to be something that is objective and measurable. Right? So let's go back to these examples and see what, and before we go there, we have to differentiate between what are primary endpoints and what are secondary endpoints, right? So you may have several objectives, but one of your objective is the main objective. And associated with that main objective is your primary endpoint. Your primary endpoint is the main thing you want to measure, right? You may be measuring 20 things, but there's one main thing that you're trying to measure in any research. And why does there have to be one main thing? Is because your study is designed to measure that main thing. Okay? How many patients are you going to recruit? How long are you going to follow up these patients? What instruments are you going to use? Those are all determined by the primary endpoint. And usually there is a single primary endpoint in a study, but there are examples where there are co-primary endpoints. There may be two endpoints, and they're both called primary endpoints, so they're co-primary endpoints. All the other endpoints in the study are known as secondary, sometimes also tertiary endpoints. But secondary endpoints are all the other things that you want to measure, but your study is not necessarily designed to measure that endpoint. We are going to measure it, but we can't declare the study successful or unsuccessful based on those endpoints. Now, endpoints like objectives depend on the phase of the clinical trial, right? So let's look at phase 1, phase 2, phase 3. In a phase 1 study, your objective was to assess safety and tolerated doses. So your endpoint is the maximum tolerated dose, the dose schedule, the pharmacokinetic and the pharmacodynamic properties. In a phase 2 clinical trial, which is trying to look at efficacy, you'll have to say that my endpoint is the primary endpoint is the objective response rate. You could also have other endpoints like the disease control rate, the toxicity rate, but generally the primary endpoint will be the response rate. In a phase 3 study, when you're comparing with standard of care, your endpoint will be survival. It could be overall survival, or it could be, progression-free survival. And you may have other endpoints, which could be quality of life. And there are many studies now where the primary endpoint is improvement of quality of life. And your other endpoints could involve toxicities. Now, these are the common phases of clinical trials. But in a phase 4 study, you basically have the same endpoints as the phase 3 study, but you're just testing them in a real life setting. Now, sometimes, even before the trial comes into phase 1, right, you may want to do a phase 0 study. And for drug trials, this could be looking at drug disposition in the body, the target inhibition rates, these are known as phase 0 trials, and the endpoints of those trials are appropriately different. Now, we are not always doing drug studies. For example, I'm not a medical oncologist. So I may be designing trials which look at radiotherapy. You may be designing trials which look at surgery, right? So when you're looking at local therapy as an endpoint, as the intervention, in phase 2 and phase 3 studies, you may have certain slightly different endpoints. You may have local regional control, pathological complete resection rate, or pathological complete response rate, functional outcomes as some of the endpoints in your study. So your study will be based on what benefit your intervention is supposed to be doing in your patient. Now, for the phase 1 studies, the endpoints are very clear. They're maximum tolerated dose, dose-limiting toxicities, and certain specifically defined parameters of pharmacokinetics and pharmacodynamics. For phase 2 trials, the endpoints are standardized response criteria. Okay, now you have to always specify what instrument you're using, or what parameter, what criteria you're using to judge a particular response rate. Okay, so you may be using the RECIST criteria, the M-RECIST criteria, I-RECIST. So depending on what investigations you're doing, or what disease you're treating, your criteria has to be specified. And generally, when you're looking at responses to a particular agent, you may want to define a complete response, a partial response, stable disease, or progressive disease. And these criteria, the RECIST or whatever, will have these definitions. I'm not going to dwell on that, right? So what do we look at in a phase 2 trial? You would look at what is known as the objective response rate in a classical phase 2 drug trial, right? So the objective response rate is the total number or total proportion of patients who either had a complete or a partial response. Okay. Now sometimes, especially in newer studies which are looking at immunotherapy, where sometimes the disease does not reduce in size, yeah, you might have an endpoint which is known as the clinical benefit rate, which is either a complete response or a partial response or stable disease. So anything which shows that the disease is not progressing. Now this is sometimes a contentious one. People could argue, you could argue that, if my disease is stable and continues to bother the patient with the same side effects in an advanced disease setting, is it really a clinical benefit rate? Now, when you are designing your trials or when you are reading other people's trials or critiquing them, you have to think from the perspective of the patient, right? So when you see an endpoint which is known as the clinical benefit rate, you really have to see that, is it meaningful to consider this endpoint in a very severely symptomatic metastatic situation, for example, or local regionally advanced situation. So you have to ask yourself, is it a good endpoint to use? Is it meaningful for the patients? Now, of course, trial design has become much more complex than it used to be. So what we are discussing are the classical designs of response criteria as the main endpoint of phase 2 trials. But many modern phase 2 trials often have survival related criteria even in the phase 2 setting, right? So you could have a progression-free survival or an event-free survival as the primary endpoint of a phase 2 trial as well, right? This is usually done in situations where progression or event will happen reasonably fast, okay, fast-growing, bad prognosis diseases, but this is what they could be primary endpoints of phase 2 trials. Now, when we come to phase 3 trials, you really have to ask some very difficult questions, right? These trials will determine whether a drug will get licensed or not, or a treatment will become a standard treatment or not. So here, you can't cut corners, right? What is it that really matters to patients? Do the patients really care 2 lung nodules or 3 lung nodules, or whether their nodules reduce from size to 3 centimeters in size? Most of the time, they don't care about it. Okay. What they do care about is living longer or living better. And it's widely understood that the most appropriate endpoints in a phase 3 clinical trial are overall survival, which tests whether somebody lives longer or not, or quality of life, the patient-reported quality of life, which determines whether they're living better or not. So these are considered to be classically the most appropriate endpoints in phase 3 clinical trials. And everything else is considered technically as a surrogate. When we say whether the disease is progressing or not, I am saying that progression is the surrogate for survival. That person who progresses is likely to die. Good salvage therapy, it may take 15, 20 years of follow-up for you to really make an assessment of overall survival. The second, and that leads to a situation where the number of events in overall survival, an event is a death, in a particular time period is going to be low. And therefore, from the investigator's point of view, and especially from the sponsor's point of view, more patients requiring more time means more money. It may be extremely expensive to run a 2,000, 3,000 patient randomized control trial for 10, 15, 20 years. It's really, really expensive. And, many people, so that's the practical point of view. And the other practical point of view is that overall survival is influenced by subsequent therapy. So even if, your new agent leads to later progression, delays progression, or delays disease recurrence, that effect may be offset by good, salvage treatments that may be available for a particular disease. And therefore, if you carefully look at all the trials in the NEJM that you have read in the last several years, a large proportion of them do not have overall survival as their primary endpoint, or not a good thing. There's a caveat. The caveat is that it ignores death due to direct toxicity or indirect effect on other organs. Now, what we forget in our excitement as researchers is that any new agent or any change to a particular protocol can have direct and indirect effects. And some of these effects can either lead to death or an increased possibility of death. You can give a treatment that over a course of time leads to heart failure. You can give a treatment that can lead to severe neutropenic sepsis. And therefore, if you're only measuring death due to disease, you're not going to account for all these things which are very, very important for patients. It's their life. Now, what are the most common surrogate endpoints that are used? In the curative or the adjuvant setting, the endpoint that is used is known as disease-free survival or relapse-free survival. And basically, it counts recurrence or relapse as an event or death as an event. So if either the patient recurs or the patient dies, it's considered to be an event, right? And needs to be marked out. In an advanced metastatic setting, the most common surrogate endpoint is progression-free survival. So you are measuring either progression or you're measuring death. Either of them counts as an event. And third is a much more flexible surrogate endpoint, which is known as event-free survival, and you are free from the menu card to choose what you define as an event. So you can have a combination of different things, all of which are considered to be an event. Okay? Now, these could be disease-defined events, it could be biomarker-defined events, function-defined events, treatment-defined events, and the examples would be that you may decide that a fracture is an event, right? In a bone metastatic setting. You can decide that PSA or biochemical failure in prostate cancer is an event, so that's a biomarker-defined event. You can decide that function is an event. For example, having to do a laryngectomy is an event, so you can define a laryngectomy-free survival as an endpoint. Or you can have a treatment-defined event. Does this person need palliative radiotherapy? Then that is an event, So, and you can mix and match and, create a jumble of several types of events and call them all events in a particular study, right? So that makes things more flexible. And one of the main reasons why it is used is to increase the chance of, from the mathematical or the statistical point of view, increase the possibility of having events so that you can count events. Why that is important is when, I'm sure you'll be taught that when you're designing a study in terms of its sample size, you need to have a minimum number of events before you can start, analyzing it. So if you just kept death as an event, then it may not occur for a very long time, and you've got a very long, expensive study. So often, event-free survival is used to, because the investigator feels that these events are important for the patient, but also sometimes simply for the statistical benefit that it provides. Now, when you're looking at event-free survival, you have to understand one thing very clearly, is that you have to question yourself. as to are all these events that are possible in a particular study of the same importance. I'll give you a real life example. Say, suppose I say that PSA increase is an event in a prostate cancer study, and I say biochemical recurrence-free survival is what I'm trying to see as an endpoint, then I am looking at two types of events: biochemical failure, an increase in the PSA, or death. Now, if you think deeply about this, the two are of completely different importance to the patient. The PSA increasing makes absolutely zero difference to the patient. The patient will be asymptomatic, just a biomarker. Whereas death is obviously very important to patients. So when you mix and match and say create an event-free survival event, endpoint, sometimes you're being really silly. And you have to appreciate, what is important when you're defining, event-free survival as one of your endpoints. Now, as I mentioned, skeletal-related events are an often used, surrogate endpoint. That's an event-free survival where you've got a pathological fracture is an event, a spinal cord compression is an event, a necessity for radiation to bone is an event, right? You can have an operability or organ preservation endpoint where resectability is rate, or laryngectomy or abdominal perineal resections. These could be events in your trial. But you have to make sure that these represent something clinically meaningful for patients. Now we come to the next question as to, okay, we've chosen a surrogate endpoint because we want this study to be practical. Are these surrogate endpoints actually good surrogates for death? Because overall survival is really my main goal, and is progression a good surrogate for death? Now, some people have, do research to see whether, something like this happens. And many of you may have heard of Vishal Ruali and some of his colleagues. And this is a paper by him, and it's evaluating the evidence behind surrogate measures as supporting approval of cancer drugs. And they have found that most of the time, surrogate endpoints do not correlate with survival. And the only surrogate endpoint that in multiple such papers and studies where sometimes it is a good surrogate for death is progression. And that is not all the time, but some of the time. And even, as I mentioned, PFS, progression-free survival, is also not always a good surrogate, because progression has limited meaning to the patient if it is not associated with progression of symptoms. And reducing progression, very important, at the cost of serious bothersome side effects of a new drug is not clinically meaningful to the patient. Okay, so if I'm trying to reduce biochemical failure, because that is my progression that I mentioned, and I'm giving a drug which is expensive and leads to heart failure in the long run, I'm not really helping the patient sometimes. Yeah. So time to progression is highly dependent on the timing of assessment. So when you're assessing progression, if you're doing your scans every 6 months or every 1 month, may make a big difference to what happens in your trial. And prolonged progression-free survival does not necessarily translate into increased overall survival, especially when the treatment is toxic or good salvage treatments are available. So this is something to keep in mind when you design your trial. And this is another paper which shows that response rates, duration of response, so doing a randomized controlled trial is important. When you're looking at response rates from phase 2 single-arm studies, they don't always translate into phase 3 clinical trial results. So to summarize, surrogates of overall survival, the advantages of using surrogate endpoints is that you can have a faster evaluation and approval and availability to patients. You get more events and therefore smaller trials, and you're unaffected by salvage treatments. But there are big cons, which, as I mentioned, gives the false impression of clinical benefit to the patients in many occasions, leads to bias in reporting, and the time to event can be uncertain or manipulated by what is how when you are assessing these patients. And you can have a censoring bias, which means that if your patients in the two groups are not getting censored because of the same reasons, that will lead to a bias. Now, as I mentioned, the other very important endpoint in a phase 3 trial is patient-reported outcomes or quality of life endpoints, and there are several validated PRO instruments, the ORTC QLQ, for example, or there are many such. The choice depends on the clinical questions. Your question may be specific, related to a subpart of a whole instrument. And what you have to do is define what is a clinically meaningful response criteria before your trial starts. And there are many pros and cons, and patient-reported outcome research is now much more mature than it used to be. And therefore, you really need to think and study and be better read in how these trials are analyzed and designed in order to do these trials better, to be robust for future assessment. And you should reach out to senior researchers in this domain. Finally, when we are assessing endpoints, we have to think of what associations have done about it. For example, the ESMO has a magnitude of clinical benefit scale. So for reported research, you can use this scale to see what is the real magnitude of clinical benefit, and it takes into account several different parameters to tell you whether to use a drug which is now regulatory approved in your clinical practice and what kind of benefit are you going to see. Finally, in observational studies, when you're designing an observational study, your endpoints are often related to association and correlation. And your endpoints could be an odds ratio, for example. So that is, I'm not dwelling in detail about endpoints in observational studies. So thank you very much, and I'm happy to be reached out at by email by any of you. Thank you.
Thank you, Indranil. Balu here. As always, nice and lucid. Any questions from the floor?
So thank you, Dr. Indranil, for that lovely talk. So basically what
you're classically. They're meant to see whether you got a good response rate, right? So that's the classical teaching. Now, but the demands of the industry and the demands of some practicalities have moved some phase 2 trials into the randomized setting. Chemoradiation in esophageal cancer. Okay. Now, the last prospective randomized control trial that has tested chemoradiation in esophageal cancer is 20 years old. Was done with different techniques of radiation altogether. So when I'm trying to design this clinical trial and I want to know what is the likely survival rate or in the control arm, I have no clue. Right? So how am I going to design this study? If I use the control, the response rates of the trial done 20 years back, then those results will probably not be correct. The results probably now are better. Yeah. So you may result in a faulty design, and you may a situation where your trial falls flat because of that faulty design. So what do you need to do now? So what you're going to do then is you're going to design a phase 2 clinical trial, which is randomized. So one arm is going to get the chemoradiation, and the other arm is going to get chemoradiation plus targeted therapy agent. Right? And what are you basically trying to see? That am I getting a signal? Am I getting some improvement? Which I define a priori is that if I get more than 10% benefit in this limited period of time, and my phase 2 trial is not going to be a 10-year trial, then I'm going to continue this trial often, or sometimes start a new, but mostly start a new trial, new phase 3 trial. Your 200, 300 patients that you treated in phase 2 are gone. So you're going to design an adaptive trial, which is a randomized phase 2 trial going into a randomized phase 3 trial if a certain interim criteria is met. Okay, so that's one practical situation where a phase 2 randomized. So phase 2 randomized trial are not used for regulatory approval. They usually, not always. So phase 2 randomized control trial is a practical alternative to breaking up your trial, or when you don't have good data in the control arm of the control arm of the intervention, because there's no prospective recent study. Okay, these are some common scenarios, and I'm pretty sure some of the other faculty members can give you different examples where this is used.
We'll take one more question.
You also have to realize that statistically, the phase 2 clinical trial may have some more lax, statistical criteria for success. For example, their alpha value may be a bit broader. Okay, in order to decide whether to go on to phase 3 or not. Because if you choose a 5% alpha error, type 1 error rate, you will require a larger sample size. But if you say that, okay, I'm accepting a 10% type 1 error rate, and based on this criteria, I will decide to go on to phase 3, then that is acceptable.
Thanks, Indranil. I think we will be discussing this in detail as we develop the protocols. And one more question, and then we'll break for tea for 5 minutes. Yeah.
Sir, with regards to endpoints, we see that some trials, they have PFS as one of their surrogate endpoints, and in some trials, they have duration of response. So, like, what would determine which one that we use? Like, where would PFS be better than a duration of response?
So you see, duration of response doesn't include death. Okay, so it's a trick. It's not a good idea. Yeah, because it then fails to appreciate the toxic effects of the drug.
Thank you, sir.
Right, so it's never a better endpoint than progression-free survival. So you have to take into account toxicities and toxicity-related deaths. But for some practical reasons, people let these trials through. Yeah.
Thank you, sir.
Thanks, Indranil. I think that's a very good background setting for afternoon. We would reach out to you if there are further questions through WhatsApp or whatever. And have a good day. Thank you for joining us. And over here, let's have a break for 5 minutes. Thanks. And then we will start with focus session.
to it. And to help you with that, I invite Dr. Supriya Chopra, Professor of Radiation Oncology at ACTREC. And over to you, ma'am.
Okay, good morning, everyone. And I think Indranil made a mention that somehow these talks got swapped. We should have first done introduction, background, and then gone into objectives and endpoints. So I think I'll just request you to temporarily just, mentally shift back to where Dr. Kumar Prabhash's talk was, right? Because he spoke about idea, where do ideas come for, and how we synthesize ideas to do clinical research. And now over the next 30 minutes, what I'm going to do is to talk to you about how to write introduction and background. An introduction and background can be very simple. So if you look into journal papers, it'll just be one paragraph and you can say, what is the big deal, right? You can just have a summary of different articles on the idea that you have and just come up with a one paragraph background and then directly go into your protocol. But no, I think here there is a lot of mental clarity needed because background and introduction is actually a lot of preparation for what you do. And while I talk about it, I will give you some examples of how we do background and introduction for some of the work that we've been doing. And my perspective that I present to you is based on doing various phases of clinical trials, phase 1 to 3, including some translational research, but also working in a more international and global environment where trials are designed for multi-institutional setting rather than a single investigator setting. So first of all, because as Balu has said, now we go into real protocol development. So what is a study protocol? It is not your science. It is also a legal document. Study protocol is a legal document. The reason is that whenever you're doing a study protocol, it goes to the Institutional Ethics Committee, which has all members. They are your framework. They set a framework for you with which you will conduct the research. You will abide by those rules. And if you don't abide by those rules, you can be penalized as an investigator. So for doing research, it does, it is a legal document. And if research has gone bad, we also know that some people are debarred from doing research, or an investigator malpractice can also come. So you have to take study document preparation extremely seriously, even if it is simplest of study. And by various components of it, I think we will do step by step. And I'm taking the first section. So we've spoken about big idea and Dr. Kumar introduced. What you have to remember is that what you write today, the protocol eventually will start after a year, because it doesn't really start in 10 days or 15 days. Any research protocol takes time to start. And whatever you do will remain active for 3 to 7 years. And if these are long observational studies, maybe 10 years. So you have to make sure that it is value for time for all investigators, because it's not a single document in which you will engage. Multiple other people will engage. So we do it in a way that everyone benefits. And generally, the expectation is that you are a very strong academician. You keep on reading books and articles, and one fine day you come up with an idea and you say, okay, I want to do this kind of research. But in real life, it's exactly the opposite, right? You may be working in a lab, in a clinic, or somewhere, and then you have that aha moment in the sense, oh my God, like you will say, okay, this is something that is not answered. Or you keep on asking, your mentor, suppose Vikram is your mentor, and you keep on asking him, tell me this, and every time he gives you a vague answer, right? Then you understand that, okay, there is no answer, we need to investigate. Maybe he's fooling around, or Supriya is fooling around, and that forms the basis of a research question, because you've tried that he'll answer, but he did not answer properly. So that could be a potential question. Sorry, Vikram, I was having fun. So that is how the research idea can come. And when this research idea comes, you may not have done preparation. So you have to do a lot of reading to make sure that your idea that you have is taken towards test of efficacy, whether your idea is effective and worth testing, whether it is relevant, And if it is relevant, will it have utility, and that too in an unbiased way, because it's your idea. You may feel to prove that right, but I may just think it is an absolutely idiotic idea, right? So unbiased way is extremely important. So now I'm going to dissect for all of you. I think all of us say in one breath, background and introduction. It's not the same. Background is different, right? And introduction is different. So for example, I say, this is Dr. Balu Nair, right? That's the introduction. But I have spent 10 days finding who he is, right? Because I have to introduce him. So that is the background, right? The preparation that I have done before calling him and inviting him in this workshop, right? So that's the difference between background and introduction. And a good background, so if I have read all his publication, it allows me to introduce him well in one paragraph, that he's professor, he's done esteemed things. So background is different. And then when you give a good introduction, it provides you a rationale for your research study, and also it helps you to build a hypothesis. So all these words are very important. Background is the work that you do to introduce someone, right? And then introduction helps you to write a hypothesis. So hypothesis is something that you want to test, right? It has a statistical connotation. Background is general, broad information. Introduction is concise, specific information. And then hypothesis is basically a statistical test where you are going to test your idea in an unbiased way. So between background and introduction, there are certain elements that when you are preparing background, you need to have some idea about what this question is, why it needs to be done, how it needs to be done. And while taking this journey from background to writing an introduction, you need to meet multiple milestones, and we will come to those milestones. So what are the essential elements, right, for background? So all of us know that when we have to write thesis or something, you prepare, you do a literature review and start writing, A to Z, everything, whatever you get from various places, because it should be 150 pages, right? That is the motivation. No, research is different. It has to be focused towards the question that you are going to ask. So first thing, when an idea comes to you, you should ask why it is needed, right? And you will do some literature search and you'll realize, okay, this is not answered. That's the reason it's needed. But there must be some existing evidence. No human on this earth, somebody said this is research, it's not search. So whenever you say research, that means someone has already done it. There's a re in between, right? So you see what is the existing evidence. Some people would have done something. So you collate that data. And then there may or may not be a gap between what is done and what is not done. So this gap between what is already done and what is not done may help you define your research question, right? That what exactly is missing and what you want to test. And of course, there is preparatory material that you will collect about feasibility of research. And so it's all literature review. So these are the essential elements. So what is the difference between an investigator A and investigator B? All investigators, everyone has similar IQ, everyone has MD degree, they start. Some people end up publishing very good research and other people will just have, simple articles for 20 years of their life. So what is important is the difference between standard and what I call as smart preparation, right? You have an idea, you may be really liking your idea that I want to do this. But I think asking for relevance is also important. And what smart investigators would do is, if I have an idea, I will go and ask a person A, I have this idea, and what do you think? And that person will say bullshit, right? That's first reality check. So you should ask 20 people, what do they think about your idea? You will get an idea of relevance, because tomorrow you're going to publish it, right? And people need to be receptive. You have to serve it to the community. And then is feasibility. So I may want to test the feasibility of driving Mercedes in Dadar, right? That's my research question, right? It's not practical. I may do a research study and say that, okay, one car rides at 11.1 and another at 12.2 kilometer per hour. But who cares, right? It's better to walk, right? So it's not, so I'm just giving some extreme examples so that you can understand. They're not really oncology, but feasibility of your research and practicality of your research. I may test, one of the most expensive immunotherapy for patient tolerability, but my patients cannot afford it. It's meaningless, right? And then applicability and cost. Again, Sachin Tendulkar gets Ferrari gifted in Bombay, right? And applicability, can he bring it, right? No. So it is not applicable. This may be good. Giving Ferrari in Europe and Switzerland is very good, but here it's not good. So applicability and cost of your research. And then later on, availability. I proved that Mercedes can drive at 20 kilometer per hour in Kharkar, right? But is it available? Can a common man afford it? That research is useless to India if I do a research on how fast can Mercedes drive in Kharkar. It's not relevant. So I should not be investing, right? People will dump it in dustbin. Why? Because India cannot afford it, and to West, it's not relevant. So these are the things that you should do. So why I'm giving these examples? Okay. So why I'm giving these examples is that these are parts of smart preparation, because your research that you're doing, you are passionate about it, but it needs to be relevant. And though examples that I've given are from real life, just apply to any research question that you're having and ask yourself once or twice. So peer review is very important, and you need to future proof and time proof the research. So for example, you want to test, I have an excellent software for data analytics, right? And I design a very good study if it can do AI analysis or not, and design a study which will run for 4 years. It is not a time proof study. Why? Because the technology is moving so fast that that software will be irrelevant after 4 years. So whenever you are designing, you have to always ask yourself this question, that what I'm doing today, I'm going to publish after 5 years. Will it still be a relevant question then? So future proofing and time proofing your research, because you may be having an excellent idea, in 5 years that idea becomes irrelevant, right? So these are the questions, and always look at failed protocols or negative studies. If they have been published, they'll teach you a lot why you should not do or where other people failed. And now we move into, I have given you some, broad introduction, which was slightly entertaining, if I may say, to just give you an idea about what needs to be done. But now you have to follow a particular rigor. And these are Medical Research Council guidelines on developing background. These are guidelines on developing background. And it recommends that you must have a systematic review of literature before you start a protocol. You should introduce your question appropriately. And if you are going to use any particular intervention can be sleep, intervention can be analgesic, intervention can be a chemotherapy drug, you need to know its mechanism properly. And then, of course, good scientists will always have, good scientists will always have some feasibility data, because if you have a research, you have to go to funding agency. You may write very good 1000 pages, but do you have experience in doing this? Why should I put my money to you? So some kind of pilot data in background always helps, because tomorrow when you take it to funding agency, they would know that you have know-how to address this. And sorry, coming back to, again, I have shown you MRC. Now more and more agencies, this is a busy slide, but what I'm trying to say is that from journey from 1830 to 2010, more and more of funding agencies are mandating that a clinical protocol must be preceded by a systematic review. Either you should have done it or you quote literature for that, right? But that is essential, because they don't want to waste time on background. If you know that there is a systematic review, and if you have done it, it gives them synthesized information about what is done. And even though there can be randomized trials on topics, still a high quality meta-analysis of good quality randomized trials still supersedes that. And having systematic review wherever possible is desirable. So how to perform systematic review? You will say, I have idea, I want to write protocol, I don't want to waste time in doing systematic review, it will delay me. But it is very, very efficient to do that. Believe me, a good systematic fast review can be as short as 3 months or 4 months if you don't want to put statistical numbers to it. And depending on the type of study that you want to design, whether it's an effectiveness study, experiential study, cost economic study, you can do a systematic search just focusing on 4 items. What is the population? This is the famous PICO model. Population, that you're going to study, intervention that you're going to do, do you have any comparator, and what is the outcome that you're testing? And depending on the type of study you are planning, so for example, if you're planning a study on a diagnostic test, these search items will just change, and you'll get these slides. I'm not going to go into details with this, but this is what systematic search for your research question should involve. And I will give you one example, that in Tata Hospital for cervical cancer, we did a very famous trial on neoadjuvant chemotherapy, right? We did not go and do a systematic search. We just relied on whatever was published. You see, in 2010, 2011, somebody had already done a systematic review which clearly demonstrated that we are not sure if neoadjuvant chemotherapy and radical hysterectomy are superior to radiotherapy alone. They could be superior, but there was no chemoradiotherapy. And this borderline information formed the basis of doing a randomized trial at our institution, which provided a level 1 evidence, because we did a randomized trial of NACT plus surgery versus chemoradiotherapy that was missing in the literature. And this has become one of the most highly cited papers from our institution for cervical cancer. So this is an example of how you can also utilize what is already published and you don't waste further time. Then another good example. So there is a lot of emphasis now on giving advanced technology treatments to patients, and you want to do a health implementation study. When I say health implementation, means that there is this new treatment that has become available and you want to make it available to your population. So when you go to guidelines, what do people ask? Is there level 1 evidence to do this intervention? But for example, if I don't have level 1 evidence, then these are the scenarios where I may have to go. And this is a meta-analysis that we did before starting a healthcare implementation project, because I wanted to start a healthcare implementation project of advanced radiotherapy techniques in gynec cancer. Tomorrow, any funders can ask, you are starting a healthcare implementation project. Do we have level 1 evidence? No. So I had to perform this meta-analysis to first demonstrate that this is efficacious, because nothing is available at level 1 in the world literature. And of course, then we had to do some, I had said, cost efficacy or feasibility studies. We will not go into details of this, but I'm trying to give you an example that before you put full implementation protocols which go out in the community, and for an intervention which is expensive, you have to do a lot of background work. Background is the word that I'm using. Introduction nahi use kara hai. Background is the word. So this is all goes into background preparation, where you create certain models of why it should be done. And if done, this will be cost effective. Otherwise, government will not put money. Interventions are expensive. And this is a third example of how you can create background. We are still talking of systematic reviews, is rapid reviews. So there are agencies, you can pay them and get rapid reviews done. All of us don't have that kind of money, but this became very relevant during COVID time, because there is an international healthcare emergency. There are so many new drugs in the market. They have to be started immediately. So what is the evidence? So many of these professional agencies would do a rapid review, and that would be commissioned so that immediately a fresh protocol can be written and you don't waste 3 months in writing up the protocol. So these are rapid reviews. Third, different from these studies, nowadays it's very, very common to come up with studies. Please notice that I'm touching different study designs. In first slide, I showed you background for randomized trial. Second was for a healthcare implementation study. Third is now pharma is very keen, and investigators to have platform studies, umbrella studies, and basket trials. So here it is very important that background is very different, because in umbrella, platform, and basket trials, you will keep on introducing a new drug and a new intervention. So here, always the background and introduction will be kept live, which means that the moment you want to introduce a new drug, you will have to update all your background information. So for platform, umbrella, and basket studies, it is a continued process of revisiting introduction the moment you get under your umbrella a new drug or an intervention. I will just wait for a minute so that we can take some real examples. But if there are any questions, I'm happy to take. If there are 1 or 2 questions now before we go into actual examples. Okay, there are no questions. Can I presume everyone has understood everything? Okay. Okay. So shall we go to some examples, real life examples? I'm going to take a very simple case scenario of a real life example of a study that I'm doing. And we had a clinical problem. I'm actually posing all of you this question. We wanted to see that if there is a patient with metastatic cervix cancer, many people use radiation as a treatment, which is a local treatment. When disease has metastasized, local treatments may or may not have benefit, but I was very convinced that it will help patients. So the idea that I had was that addition of radiation to oligometastatic cervical cancer will improve survival. That is my idea. And why I wanted to ask this question was because cervix cancer was poorly represented in clinical trials. And for cervix cancer, this occurrence is very rare, like just 10 to 15% patients will develop metastasis as compared to breast cancer, colorectal cancer, and other cancers where this incidence is very high. Then I looked into world literature, and there were only small isolated series of 10, 20, 50 patients published from places. And by doing literature review, I realized that clinical practice was very heterogeneous. Furthermore, it was not clear what constitutes oligometastatic and oligorecurrent disease, and impact of radiation intervention was not known. So what kind of study should I design? Anyone? So there are options here. So I've given you a background. I have a big idea, right? But background information is less. And it is a relatively rare situation. It's not very common. Phase 2 study. Who wants to say phase 2? Just raise of hands. I'm getting confused. So 2 people, 3 people have said phase 2 study. And why not phase 1? Because I've not written here. Okay. And who said prospective interventional? No. Who said retrospective? Okay. There are a lot of sensible people right here. Because there is no data. If I directly go into designing an intervention study, it will be very difficult, right? But I'll come to this kind of protocol. So what I said, no information is available. I have an idea, but what people do, I don't have. So what did I do for background preparation? Because your task is background preparation. So what we did was systematic review. And you can see one of my students, he did systematic review of all published data. He made a publication in 2021. This was to design a protocol. Next thing that we did was, I was telling you these elements of smart preparedness. We did a survey of clinical practice and we sent to 22 international experts that how they will treat such cancers. And there immediately we realized that many people are saying curative intent, 64%, 36 are saying palliative intent. Then again, on another context, 69 are saying, 59 are saying palliative. So in world experts, there is no consensus. So if I design a prospective study, they will mess it up, right? So what we did was after this survey, we said, okay, everyone is confused. So first we published definitions because definitions were not published. You need to give people concepts. So we spent some time there. Then we did a pilot publication just to align outcomes to the definitions that we had proposed. And after doing all this, we again asked ourselves same questions again, that can this study be done? And the answer that I got was no, we cannot do a prospective study. So we took a step back. We just did a retrospective study. So now this is a 20-center international study that has started recruiting, where we are just gathering retrospective observational outcomes, and that is how it helps. So if I had gone into a phase 2 study, I would realize that different people are doing different interventions, and finally when I analyze my results, they can be heterogeneous and they can kill the concept. Kill the concept, it means that if people are not using radiation uniformly, I will end up demonstrating that it is not effective. So it was very important, based on the background, that I take a step back and design a retrospective observational study where we try to align people with the definitions and build them up to do a prospective study. So these 5 things that I published were background. And how does the introduction look like for this study? How should the introduction look like for this study? Introduction will be just 1 paragraph. Can I have the second file, please? Can I just open it here? Sorry. So I have shown you 5 different studies, whereas here when I write the introduction, background rationale comes, introduction will just come in 3 pages, whereas background was 5 different publications and survey. So that is what distinguishes between introduction. So introduction is a concise, maybe 300 words, 500 words of what is available. But with background preparedness, I could start a multicentric international study, which now will produce a very concrete information, right? Now we will go to next presentation. No, I need to go back to the other presentation. This is not coming. This was not visible to you. Sorry. It was visible on my slide, but not that. You can go back to the presentation. So this was retrospective observational study. Till they are sorting out, I will speak about another study. So then I went on to designing another study on immunotherapy. Immunotherapy is very famous nowadays. Everyone wants to do one immunotherapy study. But the story with cervix cancer is that 100% of the immunotherapy studies in locally advanced setting are negative. But yet, I'm very persistent. I still want to do immunotherapy study. So it is a difficult situation. There is evidence that it's not going to work. But nahin, I have a big idea. I want to do immunotherapy study only. So who's going to fund me? So you have to do a very different kind of preparation, because I am still convinced, right, that other people have not done it right. I want to do another immunotherapy study. So I will come to now giving you some example of how did we prepare background and actually got it funded in an environment where all studies are coming negative for immunotherapy. So this is the second context. Can we go back to presentation? Yeah. Yes, this is okay. So this is now the second study. I have told you that all trials are negative, but I have this idea stuck in my mind that everyone has done it in a wrong way. It's very brave that when you have evidence against you, but I thought that no, their patient selection was not proper. The immunotherapy delivery time was not proper. They used radiotherapy that was suppressing the immune system, and they did not select endpoints. So I had a lot of arguments in my mind against, the study. And of course, people kept on telling us, no, who's going to fund, right? They're already negative studies. Pharma is not going to waste money. So what we did was, again, went through the same preparedness. First, we asked people that what if we combine immunotherapy with another molecule? Will you be interested? So despite having negative trials, 38 centers said, no, we are still interested, reconfirming that I'm not the only one who still thinks like this. Then what we did was we started researching the literature again, right? Why did studies fail? Why in head neck cancer, lung cancer, all studies are coming positive, especially in lung, but why not in gynecological cancer? So what we started was also started investigating that if we have to enhance immunotherapy, what are the other drugs that we can use? So we did a systematic review of the entire repurposed, this is a redo, database. We searched for all drugs that are available. So that came as one publication. Then we did meta-analysis of all available clinical trials. We published a meta-analysis. Then third thing was, in my own clinical process and lab, we had multiple time point biopsies of patients. So we started testing PD-L1 in reference to radiotherapy and seeing. So this is the pilot data that I produced, that there is expression of PD-L1 even after radiotherapy. Then we started looking at how radiotherapy impacts immune suppression. And we did some pilot work in our clinic, because I have these 5 ideas why it failed. So we started producing pilot data for those contexts. And then what we did was, because I thought their endpoints were not clear, I had a problem with the RECIST interpretation for these clinical trials. So we went through and put through our data, RECIST 1.1 versus clinical radiological response. And actually this came just online 2 days ago, where we tried to demonstrate that the endpoints that people were using were not okay, because you can just create a difference in outcomes based on choice of endpoints. So because I had to go in a different direction than was already proven, I have to do good background preparation. And these 5 publications form the background of a phase 2 single arm study, because after negative studies, no one will fund again a randomized trial. So we put a phase 2 study in which we select population properly, because I had thought population was not proper. We tried to introduce intervention at a time where immune suppression is less, and then time according to patient tolerability. This was a phase 2 study, and immediately I spent like 3 years preparing this study, and the moment it's ready to go, we have a positive study that gets published in literature. So whatever background that I've done goes flat, because now a positive study has come. So very rapidly in last month, I had to again revise my background and come up with next background, and this study is now fully funded by Merck because they found good rationale in it. Because we ended up proving that there was a reason these studies did not test out to be positive, and if you do it in our way, possibly study will be uh, successful. So what I want to show you before we summarize and end, can I have was it showing, can you see this on this slide? Okay, so this is now the full protocol of a phase 2 study, which is funded by pharma. So here you can see introduction and rationale is extremely long as compared to previous protocol where I said 500 words, because here I am going in a different direction than what the world knows. And here it is a 13-page introduction touching on each of the topics, that is background information, pharmaceutical information, preclinical and clinical studies, how do you sequence with radiotherapy, what are the side effects that you would expect, and what. So there is a full detail. So depending on context, your background can change. And now with this thing, I will like to come back, sorry, to my presentation and end this talk by summarizing. Sorry, can we go back to PowerPoint? Yeah. By saying that for any protocol writing, smart preparation is needed. It is just not, mix and match of words and taking few things from here. You need robust estimates. You will spend 5-7 years. You don't want study to go negative. Then it has to be relevant to community, and it should generate new information and interventions that will be relevant. And that will help you prepare the introduction. So I will end here. I'm not going into objectives and endpoints because Indranil already covered it, and we can take some questions. Thank you.
Thank you, Dr. Supriya. Any questions from the floor? You'll be doing this today, so it's better that you ask it now. So.
Can we have the lights on? This is actually on the.
Hello, ma'am. I'm Dr. Sabin. I just wanted to know that if you are going to do a trial or where there is no other data available, or it's a new idea or something like that, the literature is not available as of yet, like of biomarkers or some kind of preclinical studies. How do you put up a background on that kind of situation?
Okay. I would again like to say mostly for clinical contexts, I'm yet to come across a clinical context where there is no background. You can still find a case report, some kind of background thing. But preclinical is really where no data is available. That is the zone where no data is available. And that is the reason most of this stuff is done in animal studies and not human studies. And for that, even if you are motivated to test a drug, right, it may have some data in another cancer type. Like, for example, we started with lenvatinib in cervix cancer. There was no data, but we did have data in hepatocellular cancer, thyroid cancer, and other type of cancers. So we extrapolated that data in the background. I don't think we may feel that all of us are, really having brilliant ideas, But trust me, most of our ideas are repetitive or a combination of ideas that have come from different direction. And if you cannot find for the context, there may be a parallel context. So for example, you may have no data in head and neck squamous CA, but you may find in lung squamous CA. It can happen that what you are wanting to test, for that specific thing, there is zero data. But your brain has probably acquired that data looking somewhere else. So try to get that as background information, and then you take even a step back, like you're saying, for preclinical, and that should be your starting point.
Should the introduction cover the secondary endpoints or the objectives that we are looking at?
So introduction actually rarely covers the objectives. So introduction covers what is available till date, and it brings you up to hypothesis, right? That what you need to test. Secondary objectives are, most of the times are additional things that you may want to see. You can mention them, but guiding forces that we don't know, for example, if overall survival is improved. But when you are saying that drug A, we don't know if it improves overall survival, you may want to study response rate, but you're not going to really, put everything, all of the secondary objectives in your introduction. But if you think those secondary endpoints can be very, very important, for example, if they help you with response prediction or secondary objective may be biomarker driven response, then you may introduce that topic in this thing. So basically you have to see how relevant your secondary endpoints are. Some people write like 20 secondary endpoints, right? Then you cannot have rational for all. But if you have 2 or 3 which are very pressing, for example, from patient's quality of life perspective or translational research perspective, then you can devote a section in introduction for that aspect also. Yes.
So basically, I would say the same study which you said, that in the role of radiotherapy in a oligometastatic cervical cancer. Now, although we do not have that background data in cervical cancer, but we have some data in some other cancers. Let us say the improvement in outcome with incorporation of radiotherapy. Now, would that data in other cancers also serve as a sound background for conducting this research in cervical cancer?
It will serve as a background, but not sound background. Why? Because every disease has different biology, right? Some diseases may have very effective lines of chemotherapy, which may do as good a job as a local intervention would do. For cervix cancer, unfortunately, number of lines of chemotherapy are not as well established as for lung cancer or breast cancer. So the question's relevance does not depend only on that intervention, but also on what other interventions are available. So people do that in clinical practice, but ideally for every cancer type. That is the reason that if you license a drug for one cancer type, not necessarily it gets licensed for another cancer type.
Correct.
You have to really test in them.
Yeah, right. So it is always preferable to get background data for the question which on the cancer on which you are going to do research, rather than to take data from all other cancers, right?
Yes, but if they are, for example, squamous CA cervix, I can take from squamous head neck.
Got it.
This is
Some
HPV
homogeneity.
driven.
Okay,
I can
got
take from another HPV driven, but I cannot be taking data from melanoma and putting it in cervix cancer, because immunotherapy works in melanoma. I cannot use it as a background for cervix cancer.
Got it. Thank you.
There's
So I
one
think-
question here. Somebody raised their hand, or
Anyone else? Yeah.
it's just.
Ma'am, just that when there is evidence in animal studies for high-risk groups, in an introduction and a background, how are we to write it convincingly as to planning a study of such a sort where high-risk, like pediatrics or maybe immunocompromised patients are involved? So,
can you please repeat your question? What do you mean by high risk?
Pediatrics or immunocompromised patients, if they are the ones who are to be put to test for a drug which has only been approved, say, in animal studies. So, in our background, how can we make a convincing argument for that?
You cannot go. Actually, in vulnerable population, you're talking. So these both populations are vulnerable, whether it's immunocompromised population or it is like they are not vulnerable by definition, but they are high risk, right? Like pediatrics and this thing. So you cannot use a drug directly. You will have to go through rigors of phase 1, phase 2, and phase 3 before you use. In end of life situations, if you want to do a protocol, that is easier because getting approval to do testing there is fine. So if you want to go from animal to phase 1, then actually rather than a population that's going to live, a population that is terminally ill is more suitable. And that's the reason most of the drugs are first tested in metastatic setting or terminally ill. So if you have animal data and you have to go into phase 1, then population should be chosen accordingly and not population which is expected to live long or expected to have high degree of complication like immunocompromised patient. No.
I have gathered all the information now. Whatever the information is needed for a background, I have gathered. So how to craft a background? Like what type of information should come first? What type of information should come next?
Yeah. So how much time do we have, Vikram?
No, we are about 5 minutes behind schedule, but maybe we can take 2 minutes.
Okay. I will just show you two types of crafting. Coming back to this immunotherapy study, you've seen this. I will go section by section, right? Here, I have said I have invested 12 pages. So let's look into how we have written introduction. So first of all, we have said that multiple studies have investigated. They are the first line standard of care. Then I have come to saying somewhere here that various studies have been done which are negative. Then I have come directly to immunotherapy studies here which are negative. And then I have come to criticizing those studies because I felt they have not been done in, a good way. And then I have come with the background of drugs that I want to test and how their combination could potentially work, right? And then towards the end, I have to now convince people, right, after this. Then, of course, I write this preclinical. So somebody was asking of other cancers. So here I have spoken about NSCLC, RC, UV, melanoma, right, what these studies are. But the end, I have come to say why it will again work. Other trials have failed. So I have said, what are the specific strategies? And these 5 publications that I made are the 5 points here, right? I have full publication for each of the point, but they are concise. So their conclusion is a point here. And then I have introduced this, that how I will do it so that this will be better. And then, of course, listing of adverse events that you can expect in this population. There is, of course, a lot of background work. So here you can see that background is much larger and then it leads to objectives because I have to do more convincing. Whereas for the retrospective study, because I have to do, just 1 second, because I have to do less convincing, it is a low stake study. No one has studied that, right? So their background is one paragraph, and then I just put definitions, and within 2.5 pages, I come to directly objectives. So a high stake study, or if you think people will not fund, your background has to be strong. Otherwise, they will just say, go, don't waste our time, right? Don't ask for money. I may write the study, I take 2 years, but it needs to be funded, right? If I don't present a strong rationale, no one will fund it. So what you are seeking should help you make that concise statement. आपको study करना है और उसके लिए आपको पैसा चाहिए, right? That is the bottom line of research. And then you will get a publication after 3 or 4 years. So that should guide your background.
Thank you.
Okay. Okay, we'll end this. Thank you very much for your questions. Thank you.
Thank you, Dr. Supriya. Next, we will have Dr. Prabhat Bhargava from Professor of Medical Oncology, TMH, and he will be talking to you about how he approaches a protocol writing. And over to Dr. Prabhat. Online.
This is not any approach, he is trying to use AI algorithms for writing protocols. So we want to bring in that angle. And with that intention, we have designed this, we have called for this talk. Thanks.
Good morning, everyone. This is Dr. Prabhat here from Tata Hospital, Tata Memorial, consultant in medical oncology. Just that this interesting topic came around where we cannot run from AI and we have to use AI in our day-to-day activities and not completely rely on AI. So this is where the balance and how can it help in writing our protocol. That is the topic given to me today. I have made initial just few slides of PPT, and I thought it would be better if I can demonstrate everyone how to use it. So I will give you any random topic. One of my students is there attending his topic as example. How can we use in doing our background check or to help AI to write our protocol? Or assistant writing our protocol will be a better word, because now the guidelines have come and even journals check whether you have used AI, then the articles get rejected. So coming to start that. So we all know that we need to write to explain, as Dr. Supriya was also saying, that we need to do our own background search first, then write it on paper to convince others. You cannot be present everywhere to convince others that I have written a brilliant program, that my idea is very brilliant. So that has to go in your background literature. If you have not thoroughly searched your background literature, there may be studies at all examples, even I was listening to Dr. Supriya, that there may be studies which we have neglected and which would have given a negative result already, or small pilot studies maybe, and that is why we are not aware. So there are multiple topic headings under which we have to write a protocol, extensive document, and various places out of this we can take help of AI to do something which is repeatedly there in many of the protocols, rather than rewriting all of them from beginning, from scratch. So AI can help in various stages of clinical trial, AI and machine learning, like starting from protocol designing to setup, and there are companies now who help in managing all this using AI to find sites which are which have good outcome and which give patients consistently, to find such sites, to analyze data. Data analysis has become a very big thing, to manage data, and then to submit to regulatory US FDA regulation. So at various all these steps, you can use AI and machine learning in your day-to-day research. So machine learning can be applied to protocol design and language translation using existing protocol data and health libraries for specific therapeutic areas. A protocol for a new study can be generated by the system. We will see how to use this. The machine learning algorithm would be able to design an optimal protocol from the knowledge base, leading to reduced design times and protocol amendment and strategy and disruption. So before this, the AI can help you in multiple phase, that literature review and data extraction, to design how to design and optimize your protocol, what should be the ideal eligibility criteria, predictive analytics, risk assessment, adaptive trial designs, natural learning processes, data collection and monitoring, patient engagement, ethics and compliance, data analysis, and continuous learning. Looking at the increasing interest in AI of many people, the consult AI and Spirit AI guidelines have also come, where before you report, you have to say that how much use of AI you have taken in your trial protocol, and you using when you report it in the study. Let's start using practically. Is my Chrome visible? So let's start with Research Rabbit. This is a tool where you have to sign up first. Most of the AI tools, you have to sign up first to start using them.
Sorry for interruption, sir. Your Chrome is not visible.
Prabhat, 1 second.
You just
we
need
are
to
only able to
reshare
see
your
the
Chrome.
PPT. We are not able to see the Chrome
Okay.
browser. I think you opened a browser, no?
Yes.
No, we are not able to see that.
So you need to stop share and reshare
You
your
have
browser
to,
again.
yeah, stop share and reshare your whole screen, right?
Yes.
Yeah.
Is it visible now?
Yes, sir, it's visible.
I just logged into it. I already created an account on Research Rabbit. Even I found out all this. I had only used ChatGPT by myself, but thanks to the given topic, I have done extensive search in last 1 week to see how can we enhance our background search, at least reduce our manual work, in writing a research protocol. So first app that I would like to say is Research Rabbit. Research Rabbit helps you in identifying related PDFs, which are journal articles or book articles which are already there. I would request you to give one topic, or should I use my student's topic? Even I have not done a research of my student's topic using this. Let's do it in front of you.
Okay, you may use your student's topic, Pravat.
Valproate in neuropathy is the topic that I have given to him. So I have made a collection name and I would like to add papers to it. So it will ask me to find keywords, but I either directly article if you have it, or you want to do literature search, so related PDF will come. What all do you want to search? Mostly we use PubMed based. So it says, give me an option. Now it is searching what all new related articles are there. It did not find any related article. Maybe I've given very odd topic. So it found out some in peripheral neuropathy, chemotherapy induced. So articles are there. Recently, they do not, most of them start from recent ones and then go down. But yes, this article itself can be of importance to me. What all people have tried, rather than just. So I have added it to my collection. I will randomly add few collections just to hurry. Some emerging targeted therapy was tried. Yeah. So it once you have added the articles which are relevant to you, you can click them. It can give you a summary of the article here itself. If you really want to visit, if you like that article and want to visit, you can click here and you can directly go to the website of that journal. You can download a PDF from here itself. It can even give you similar work related to this article. So it gives you all the references of that article. If you like any, when you are going through the detail and you want to just do cross reference, all the references are here. And how are they related? This is something which I like. You can see how are they related and what was the latest one. If you are interested and not interested in 1989 topic, then you can click from here itself and go to the related articles. If you are interested based on the authors, that is also feasible. If you like, you know that somebody has worked on peripheral neuropathy, chemo-related peripheral neuropathy, and you want to see what all work has he done, directly it will even show you the authors. Suggested, it is showing you the authors who have done good amount of work on neuropathy. As per these two topics, these two PDFs only that we have selected. If you bring a library, that will help you much better. If you have 5-10 articles of your interest based on which you have to do your study. Another thing that I liked is you can directly get a citation of this paper from this, so that when you are referencing it, it becomes easy rather than writing and making it in the variants like APA or Vancouver style, whatever you want, it can give it to you. Another thing is that you can share it. Like if my student that whom I have sent, if I were to share the collection of articles, then also I can do it. I am not able to. So if I want to share it, then I can just write the email of my student and invite him. Then as like in Google Drive, you have a Google Drive where you're working on some papers and you share it. Google Drive, most of you would have used, and you regularly share it with each other. Here you can directly invite your student or your colleague whoever is working. Many of the people have made multiple apps. Like for same thing, you have thousands of apps in your mobile. Similarly, for this, you have many apps. I like Research Rabbit for one reason, because it is free for life, is what they have written. somewhere, which I saw. And people have compared it to Spotify for papers that you can track it and keep it in your library till whenever you want to delete the whole library together once your project is completed. Next important website or AI app that I like is Skyscape. SciSpace. Here I have randomly written for study that I had used using ChatGPT, trastuzumab emtansine in biliary tract cancers. So as soon as I wrote it, it has given me a summary of, in short, from the top 5 papers. But when I was doing it in last week for the first time, it mistakenly used, and rather than TDM1, it gave me example of TDX. So you have to be sure that what it is saying is right or wrong. If there is no relevant, because trastuzumab emtansine trial we are doing since last year, before that there was no evidence of trastuzumab emtansine in biliary tract. Okay, so AI is not that intelligent that it will give you only relevant article. It can give you anything rubbish. It has to fill up the number of words. It does fill up. So you have to check really whether it is useful or rubbish. Then it has given you articles from where it has taken this summary, post first 5 articles. You can read these articles again by just the insights, because this is a case study only. There was a small insight over here, just a case study of trastuzumab emtansine in second line biliary tract. So just one insight. Otherwise, most of them would be review of literature, because there was no much evidence of TDM1. Now TDX, there is a study, so it has quoted that study also. You can ask Copilot. There is Copilot is where you can use this to enhance. If I want to read the study or ask, I will just copy this. And ask it to give me details about this article. So it has inbuilt, and it can give you an idea whether should I read this article or it is not at all related to me. So it has given you, it has given to me intention only. See, this is what happens, or maybe I have not copy pasted it right. So it has given you in short what was the study design. It was a part of the My Pathway 2 study, phase 2 single arm study, where treatment approach was to see the efficacy of the combination of trastuzumab and pertuzumab in HER2 positive patients. Rationale is that those who are overexpressed, obviously it measures, and this were their outcome measures. So in short, it has given you an idea that yes, this may be one of the study which I can look after, because it has also similar cohort of HER2 positive patients in biliary tract, and they have tried pertuzumab trastuzumab as a single arm study, and my study idea is almost the same. So you can download this study further if you like the idea. So this app helps you to find out in short, and you can add what all things. You can download PDF. You can add columns whether you want only results. What all would you like to have in short over here? Who were the contributing authors if you are wanting to see relevant wise? What were the limitation of studies? Why were they unsuccessful? Or other things. Conclusions I have already selected, so conclusions are coming on the right hand side of the screen. I will remove PLDR. My computer is overloaded because I've kept so many open so that even I don't have to open it. I will close Research Rabbit. Okay, now only directly conclusion. So you can modify what all relevant things you want, and accordingly it will help you. It can give you all open access articles only. If you want only open access articles, you can download PDF from here itself. And one important thing, a good thing that I like, you can add a Chrome extension of it. I have enabled a Chrome extension of it. So on right hand side of my screen, whether you are able to see, you can see the symbol over here, a cursor, launch size page. So if I want to read something, if this is, I will come to this example later on, what this explain paper is, just giving you idea. As soon as I select this and I right click, I can directly go to this extension on right hand side and select that I want to in on the XYZ side. For that reasons, I click one more tab. Just suppose I want to read this paper. I have downloaded so many AI that my computer has become slow. I will delete it. Okay. Here itself, you see it has come. Now I can ask directly to size page, explain me this. I was trying all of them and I have not removed them. Here. So it has gone to this and it will explain me in short a summary here itself. So anywhere, any website you like, because of the Chrome extension, you get a mark over there itself and it can give you an idea. Now you want further, you can ask a follow-up question. You have a specific doubt about this paper, you can ask directly ask. Was Mirtazappan really helpful in increasing appetite? I have not gone through the paper, just asking it, was it really helpful? Let's see what the AI says. So Mitra did not show a significant decrease in appetite score. So maybe it was a negative track. However, the Mirtazapine group had a significant increase in energy intake over 4 to 8 weeks follow-up period, particularly in fat intake, which is crucial for energy. The addition of mirtazapine in the treatment of patients with NSCLC and anorexia may help in achieving their energy requirement and improve health related. So you know that what are the key takeaway from this message, from this paper, and you need not read it if you have crunch of time. It's better if you read complete paper, but if you have crunch of time and you have to review of literature of many hundred of PDF you find who are just phase 1, phase 2 trials and you want to plan a phase 3 trial, then this can give you a summary. From the PDF of this, I am just on NCBI website. I did not even download the PDF. Okay. So that was for size space. I would recommend second, after listening to so many videos and doing literature since last week, I liked size space is one thing which I liked. Maybe again, this has a library. You can add to your Zotero also from here itself. So it will keep citation in your Zotero or Medley, and then you can use that directly while writing the paper. Here there is an option of beta version of notebook. Maybe you can start writing your paper or thesis or research article in the website itself, and it will keep it with itself. There is citation generator also, because you can choose what way you want, APA or MLP or Vancouver style. It will directly generate citation from the PDFs that you have selected. Good thing is paraphraser. If you have written something, you can ask them, if I have written a statement, just casually I can write a statement of trastuzumab contents in something is not useful for biliary tract. And I ask him to write it in the academic format. In academic format, maybe efficacious word or some other word should have been come. So it will paraphrase it, demonstrate limited efficacy. So you can write casual statements, your idea, whatever you have in your mind, and then this can help you in making it official academic statements. You can increase the length of the statement. If there was a paragraph supposedly on the left side, you can ask them to reduce the length of the paragraph. If you have word constraints, supposedly you want to send us abstract of your paper and there is the word limit of only 300 and you are not able to reduce, then maybe you can ask the AI tool to reduce the number of words by using this length variability option and paraphrase it to look so that you can write in your own language easily and then it can look more academic. This is one useful part that it has an AI detector in itself. Before you are sending it to for publication or submitting that abstract, you can check really how much of your article has AI. So that will help you finding and refine your protocol and it can look more human. Later, if you copy paste any Word document that you have, you can do that also here itself. And there is a link for Chrome extension which I have already downloaded and added, so we could do that swiftly. Now let's come to ChatGPT. The first thing that I should have done, but I like to show you a background check first. Okay. Now ChatGPT also can give you complete protocol, but ChatGPT is the first basic AI that came to you. What I will say in normal words, it is a very intelligent friend whom you are meeting after many long time. Now he doesn't know what you do, and you know you have to take his views. Okay, so if you just write down trastuzumab M10sin in biliary tract, it will give you anything, and it will may not be of use. Okay, let's see what it is saying. It is saying that trastuzumab M10sin also knows this, but because there is no evidence, so last statement would be for up-to-date information and guidelines. So why is he saying this? So one shortcoming of ChatGPT is it is updated only till 2021. There is no, if a trial would have come after 2021, it is not searching it. So for that reason, there is a web extension. Another web extension, one I showed is space. Second one is extension. I had kept it off right now to show this only that there are lacunae. So there is an extension called Web Chat GPT. If you switch it on, then it will search web. Now if I update it, see, now there is web access. So now my search will be up to date. So just don't go to Chat GPT. It's better you evaluate, first add on the extension and then you write it again. This one is. So it is understanding question, it is browsing the web also, and then it will generate now. So it will take time, but it will generate a more elaborate one. It has even started giving me sources because now it is on web. It is searching on web and it is giving me references also. It has given me 5 references. So just ChatGPT doesn't help. Giving it web access will enhance your search. You can see how many search results you want. If you want to write all a big paper and it wants me to sign up, I will not taken up the paid version till now. Okay. Let me come to one example of this. So this is the protocol that I actually used, took help, and it gives you old history also. So I asked him, help me in writing a phase 2 trial. It gave me something. But this friend, it's now, that is why I used, this friend is not aware who am I, whether I am a layman person now, or I am a doctor, or I am a researcher. Then I had to give him a title. No, I want to write a study. Then it gave me in form of study, including intervention, study design, ethical consider data collection and monitoring analysis. But this is just a synopsis that I got. Then I sent him a synopsis to I have made. Then I gave him synopsis. The synopsis which I had already made. I was just checking it out. Okay, I had already written a protocol. Okay. Then it says, oh great, your study synopsis look well enough. It gave me some suggestion. Okay. Then I asked him, if I have a synopsis, can you write me what do you think is the brief management of advanced or metastatic biliary tract in second line? So essentially, this will become my background, that what is presently a second line. So it gave me something, and that is actually true. There is only one trial of HER2 in second line, of adding trastuzumab to Folfox, and there is baseline that ABC06 which says that Folfox and ABC. So you have to feed him at individual point, and then it will give you an idea. And okay, who are you? What is your level of understanding? Accordingly, the ChatGPT will give you the answers. When he understood that I can understand and I need it in more detail, then he's giving me in more details. So then I asked him the role of TDM1 in second line HER2 positive breast cancer just. Then he gave me trials in breast cancer. Then I wanted whether what are the options in HER2 positive advanced biliary tract. It gave me options, but it started writing M10, which is false. So you also have to see that we saw initially in today's SciScape also that there were only case reports for trastuzumab M10. So there cannot be a data. Okay? But because I gave him data that I wanted to trial, it assumes that trial is already is being studied in clinical trials in advanced BTC. Then I asked him deruxtecan regarding deruxtecan. So deruxtecan, there is one trial has already been published in second line. So then it again gave me something rubbish, not related to my topic. So I had to re-specify in biliary tract. Then it gave me TDX in biliary tract. So rational for my trial. But now I need not give him rational for which trial. Now he knows in what context we are talking about. So as soon as I wrote only rational for the trial, it gave me a rational for TDM1 in biliary tract. And this time it would be relevant to me. What will you suggest as a hypothesis for? So it now gave me a hypothesis that this M10 as second line can be used because there is only Folfox and irinotecan as standard of care till now. That Folfox plus trastuzumab is a small trial, so that is not a standard of care. So maybe TDM1 can become a standard of care. I asked him, do you know mechanism of action? It gave me something. What are the common adverse events? Just I was also checking out how. Then I tried sample size calculation. Can it give me a sample size? Sample size for the study also I tried. Whether it can give me. It suddenly gave me a number of 40 with all this explanation also written. Then when explanation was also written, it had assumed some hazard ratio. No, it did not assume using an hazard ratio. So I asked him whether the sample size based on hazard ratio can you give me? It gave some complex formula which I don't know. You will learn in statistics, and it gave me something. So you have to train it. You cannot just ask give me sample size. You have to train it using this power, using this, using that. Then it will give you. So when I was doing search for you, I realized that you have to give him keywords. So I have noted the keywords. So before just now, before coming online, I was trying this out just when Dr. Supriya was giving a lecture. So if you say, give him details of everything in concise, this is just now that I tried. Write a phase 2 single arm study of trastuzumab emtansine in biliary tract cancer as second line therapy. Act as a medical oncologist. Type of text, it is a research protocol. Style, academic. Tone, professional. Language, English US. Reader comprehension, readers would be doctors. Length of roughly 50,000 words. This now you can see if I've given him everything, and if you find any error, then you can redefine your further things that you need. Then it has given me almost complete protocol. But till that time, web access was not given because I had kept it off for demonstrating it to you. So it has not given me references. It has given me title. It has given me introduction, that what is biliary tract cancer, what is M10, what is the rationale, what should be the rationale of my study, objectives, primary objectives, secondary objectives, study design. So this can help you in many a times even I have starting block, where to start from. Once I have got where to start from, I can work on it. So maybe this can act as your skeleton, and then you can work on it and redefine it further. Don't rely directly on it. it has taken for granted it is, no, I have written it as multi-center study. I wanted to add, but I did not write. But it is taken for granted it will be a multi-center study. Study population would be 18 years of age who will be HER2 positive. It has left it blank what dose, because biliary tract intensine dose is still not there, so it has left it blank. You can fill it up and take help of it, the basic, what will be the study procedure, primary endpoint, secondary endpoint, most of the things it has given to you. Okay. This is how you can use it. And if I would have kept web access on, let's try in front of you. Now that web access is on, I will copy the command again. Okay. Again, it is browsing, and maybe it will give me another, because it will be up to date in last 2 years what all have happened in HER2 in biliary tract. That will also be updated. So it is a single arm phase 2 study. Oh, it is. Answer is completely modified. That you are asking again, maybe he wants that I want something else. That is why he has made introduction only of 2 lines now. Maybe you can write down, give me, you can modify it. Write introduction in details. It has gone completely out of field. I told you just now that if you are related to one topic, then it will give you related to that topic, but suddenly it is deviated from the topic. It has gone to what is really an introduction. So you have to give not as I also told you, there is guidelines for it, Spirit AI guidelines. I was trying to show you. So you definitely have to declare it that it is written with the help, what all help that you have taken while writing while writing your study. There are guidelines for that. And if you don't declare it, even in many journals have rejected even after publishing the article. So they had to take it back. So declaring is important. And obviously nobody would like that whole article is written by AI. There should be human touch to it, and you can take help of AI. As I was trying to say, if there is initial block, it can give you a guideline. You can then write a complete paper on it. But using completely AI, then there will be no fresh knowledge which is generated. So why will a journal be interested in taking an article?
Yeah, am I audible, Dr. Prabhat?
Yeah.
So this is a hypothetical question basically. So now we have a lot of ideas. Now many of these ideas will fail actually. So based on the existing literature, based on the existing background, if I ask AI apps whether the idea which I am putting forward, is it gonna be successful or is it gonna fail? Are we going to get anything or it's just
So while I was also exploring whether to use it or not, I will give my experience because it had no idea whether trastuzumab intensive will work in biliary tract cancer. It was not giving me a correct answer. So you have to use your justification also, or maybe I'm not in such an expert. I am also a medical oncologist, not an AI expert. But that is the barrier that I want to break through this lecture. That there will be nobody who is an AI expert and who is a doctor also. So we only have to start using it and become AI expert for our level. I also tried myself for writing first protocol as a joke only. Chalo, let's try क्या होता है AI कैसे करते हैं. Then I made it, and last 1 week only I have read all this Skyscape. In spite of using ChatGPT last year, I also did not use all those Skyscape and everything. So once you start using it, then only you will learn. Then only AI will also learn from it. But what I found out that there are even pharma companies at molecular level are trying whether valproate can really help in peripheral neuropathy. Just giving example, they are trying to use it, but I'm not aware of all those softwares at that level at molecular level.
Thanks, Prabhat. Just a question from on the side from me. We realize that when we read a publication, nobody reads the background and this thing. When a thing comes for review, probably the reviewers also maybe skip the background and things like that. So now, do you expect the PI also to read the background once the AI is going to give it to us? Who is going to read the background for a study, which Dr. Supriya has actually taught us how to write and.
Sir, I don't think it really matters to I started my talk also that if you are able to convince from your background only whether I should get funding or not.
Sure.
Yeah, I started for that.
No, it was
It's
just
very important.
just on the lighter vein. I understand the utility of these AI device tools. Generative AI, the way of asking questions, it's called prompt engineering, and it's a skill to develop, and that's what he had beautifully shown you how to ask questions and get your answer. And you use that skills and structure it the way Dr. Supriya had shown you, which will make a complete background. And that's what probably you would do this afternoon, maybe without AI help. We are cutting off the internet. Okay. Thanks, Prabhat. Thanks for detailing this to us. And let's move on to lunch. And after that, let's have a very practical session on how to develop your protocols up to what we have been describing today. Thank you.
Excuse me, everyone. Let me take 2 minutes of your time. We have a small feedback paper that will be circulated. Can you take 2 minutes to fill up and give it back to us? Just so, yeah, so that we can improve our next session.
speaker from USA who will be giving the first talk, and because of the time difference, we have to start early. So kindly note that tomorrow's session will start at 8:45 a.m. In the morning. And thank you very much for joining. Hope to see you all tomorrow. Thank you. And so the protocol development sessions, it will take place over across 3 venues. So 2 of them will be in this hall, one in this end of the room, and the other session will happen on that end of the room. And I think all of you know which group you belong to, because we have created WhatsApp groups and we have added your name, so you must be aware of which group you belong to. Only thing is, what I don't know at this point in time is where each of these groups is sitting, and that I will request Mice Ideas, Darshan from Mice Ideas to help us understand. So, but what I want to tell you is there will be 2 groups here in this hall, one at each end of the hall, and the other group will be going upstairs on the 3rd floor. I think the room is Sapphire, and there is provision for about 20 people to sit around a table and discuss the protocol. So the 3rd group will go there. So which group will be staying where? Darshan will be able to tell you.
Group 1 will be here, group 2 will be that side, and group 3 will be on the Sapphire. Sapphire will be on the 4th floor.
4th floor. Okay. Yeah. Sapphire is on the 4th floor. Anyway, we'll all have lunch here right now, and after that we can disperse to our respective breakout sessions. Thank you very much.